Lesson 1. The Cell Theory and Cell Diversity. We begin our curriculum with the absolute foundation of all biological science. Before we can understand the complex machinery of life, the firing of neurons, or the beating of a heart, we must understand the fundamental rule that governs it all. We must look to the smallest independent unit of existence. This lesson establishes the cell as the basic structural and functional unit of all living organisms. Let us begin with the concept of scale. Imagine for a moment that you are holding a cork stopper in your hand. It feels light, perhaps a bit rough. To the naked eye, it appears to be a single, solid object. But if you were to slice a remarkably thin section of that cork and place it under a powerful lens, the illusion of solidity would vanish. You would see a honeycomb of tiny, distinct chambers. This is exactly what the English scientist Robert Hooke saw in the year 1665. He looked through his primitive microscope at a sliver of cork and saw thousands of tiny, empty boxes. They reminded him of the small, simple rooms that monks lived in, which were called cells. And so, he gave them the name that we still use today, the cell. Hooke was looking at the dead walls of plant cells, but he had opened a door to a new universe. Over the next 2 centuries, other scientists would peer through that door. Matthias Schleiden, a botanist, looked at thousands of plant samples and realized that every single part of every single plant was made of these cells. A year later, Theodor Schwann, a zoologist, found the same truth in animals. Finally, in 1855, a physician named Rudolf Virchow added the final, crucial piece of the puzzle. He stated, in Latin, omnis cellula e cellula, all cells come from cells. These discoveries formed the cell theory, the bedrock upon which all of biology stands. It is a theory built upon 3 fundamental tenets. Let these 3 rules sink into your memory. First, all living things are composed of 1 or more cells. Whether it is the bacteria in the soil, the towering oak tree in the forest, or the complex tissues of your own brain, life is cellular. There is no life without the cell. Second, the cell is the basic unit of life. It is the smallest entity that retains the properties of life. A single protein is not alive. A strand of DNA is not alive. But a cell, which contains them both, is alive. It metabolizes energy. It responds to its environment. It maintains a stable internal state. Third, all new cells arise only from existing cells. Life does not spontaneously appear from nothing. It is an unbroken chain of division, stretching back billions of years. Every cell in your body right now is a direct descendant of a cell that divided a moment ago, which came from a cell before that, all the way back to the very first spark of life on this planet. Now that we understand the unity of life, we must look at its diversity. While all life is cellular, not all cells are the same. We categorize life into 2 primary domains based on the complexity of their cells, prokaryotic and eukaryotic. Let us first visualize the prokaryote. The word comes from the Greek pro, meaning before, and karyon, meaning nut or kernel, which refers to the nucleus. Prokaryotes are the simpler, more ancient form of life. They essentially lack a kernel. They have no membrane-bound nucleus to hold their genetic material. Instead, their DNA floats freely in a region called the nucleoid. They are the minimalists of the biological world. Bacteria and archaea are prokaryotes. Imagine a small, capsule-shaped room. It is efficient and uncluttered. Inside, the chemical reactions of life happen rapidly. These cells are generally much smaller than the cells in your body. Because they are simple, they can reproduce with incredible speed. A single bacterium can divide and become 2 in as little as 20 minutes. They are the most abundant life forms on Earth, thriving in the deepest oceans, the hottest deserts, and even inside your own digestive tract. Now, shift your mental image to the eukaryote. AU means true. These are the cells with a true kernel, a true nucleus. These are the cells that make up plants, animals, fungi, and protists. These are the cells that make up you. Imagine the eukaryotic cell not as a simple room, but as a vast, walled city. It is significantly larger than a prokaryote, often 10 to 100 times larger. Inside this city, there is a high degree of organization. The most defining feature is the nucleus, a large, central vault protected by a double membrane. Inside this vault, the cell keeps its most precious treasure, the DNA, organized into complex chromosomes. But the complexity does not stop at the nucleus. The space outside the nucleus, the cytoplasm, is filled with membrane-bound structures called organelles. These are like the specialized districts of the city. There are mitochondria, which act as power plants, burning fuel to create energy. There is the endoplasmic reticulum, a sprawling factory floor where proteins and lipids are manufactured. There is the Golgi apparatus, the shipping center that packages these products for export. This compartmentalization is the secret to the eukaryote's success. By separating different chemical processes into different rooms, the eukaryotic cell can perform many complex tasks simultaneously without interference. It allows for specialization. It allows for multicellularity. Because eukaryotic cells can manage so much internal complexity, they can work together to build complex organisms like a human being. Let us deepen this comparison. Consider the DNA itself. In a prokaryote, the DNA is usually a single, circular loop, like a rubber band floating in the center of the room. It is naked and accessible. In a eukaryote, the DNA is linear, forming long strands. It is wrapped tightly around spool-like proteins called histones, packed away efficiently inside the nucleus. This packaging allows eukaryotes to hold vastly more genetic information than their prokaryotic cousins. Furthermore, consider the boundaries. Both types of cells are wrapped in a plasma membrane, a flexible skin that controls what enters and exits. But many prokaryotes also have a rigid cell wall outside that membrane for protection. Some eukaryotes, like plants and fungi, also have cell walls, but animal cells, like your own, do not. Your cells are soft and flexible, allowing for movement and shape change. Let this distinction settle in your mind. Prokaryotes, small, simple, ancient, with no nucleus and free-floating DNA. Eukaryotes, large, complex, evolved, with a protective nucleus and specialized organelles. Understanding this division is not just academic trivia. It is the basis of modern medicine. When you fall ill with a bacterial infection, a doctor might prescribe an antibiotic like penicillin. Have you ever wondered how the medicine knows to kill the bacteria but leave your own cells unharmed? It is because of these structural differences. Penicillin attacks the specific machinery that builds the bacterial cell wall, a structure that your eukaryotic cells do not have. The drug targets the prokaryotic architecture, destroying the invader, while the eukaryotic city of your body remains safe. We are, in essence, a cooperative colony of trillions of eukaryotic cells. Yet, we rely on the prokaryotes. We have more bacterial cells in our gut than we have human cells in our body. We live in a symbiotic relationship with these ancient, simple organisms. Life is a tapestry woven from both threads. As we conclude this first lesson, return your focus to the single cell. Visualize it floating in the darkness of your mind, a sphere of life, a tiny universe containing order, energy, and information. It is fragile, yet it is the strongest continuous force on this planet. It is the vessel that has carried the flame of life through billions of years of history to reside, right now, within you. Let that concept settle in. Life is built of these tiny units. As we move inward, we must next understand the chemical building blocks that make these units possible. We must look at the atoms and molecules that construct the walls of the city. Our next lesson will take us down to the molecular level to explore the chemical components of the cell. Lesson 2. The chemical components of the cell. In our first lesson, we established the cell as the fundamental unit of life. We looked at the house itself. Now, we must step inside and examine the bricks. A cell is not magic. It is a machine made of matter. To understand how this machine functions, how it moves, how it thinks, how it replicates, we must understand the materials it is built from. We must descend from the biological to the chemical. This lesson explores the molecular building blocks of life, the biological macromolecules. At the heart of all life on Earth is a single, remarkable element, carbon. Life as we know it is carbon-based. You may have heard this phrase before, But let us consider what it truly means. Carbon is unique among the elements because of its versatility. A carbon atom has 4 electrons in its outer shell that are available for bonding. This means it can form 4 stable covalent bonds with other atoms, including other carbon atoms. It can form long chains, complex rings, and intricate branching structures. It is the perfect scaffolding for the complexity of life. If the cell is a city, carbon is the steel framework that holds every building together. Attached to this carbon framework are other elements, mostly hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Together, these atoms combine to form the 4 great families of organic molecules that make up every cell in your body: carbohydrates, lipids, proteins, and nucleic acids. Let us examine them one by one. First, we have the carbohydrates. When you hear this word, you might think of bread or pasta, of fuel, and indeed, at the cellular level, carbohydrates are the primary source of energy. The name itself tells you their composition: carbo for carbon and hydrate for water. They are made of carbon, hydrogen, and oxygen. The simplest carbohydrates are monosaccharides, or single sugars. The most famous of these is glucose. Glucose is the universal fuel of life. Right now, your blood is carrying glucose to your brain cells, where it will be broken down to power your thoughts. These single sugars can link together to form disaccharides, like sucrose, or long chains called polysaccharides. Consider the plant cell. It links thousands of glucose molecules together to form a stiff, rigid structure called cellulose. This is the fiber in your diet. It is the main component of plant cell walls, providing the strength for trees to grow hundreds of feet tall. In your own body, your liver links glucose molecules into a storage form called glycogen. It is a reserve tank of energy, ready to be tapped whenever you need a burst of power. Carbohydrates are both the fuel in the tank and the walls of the garage. Next, we move to the lipids. These are the fats, oils, and waxes. Unlike carbohydrates, lipids are hydrophobic. They fear water. If you mix oil and vinegar, they separate. This natural separation is the key to the lipids' most important role. In the watery environment of the body, lipids clump together. The cell uses this property to build its boundaries. The membrane that surrounds every cell, the skin that defines where the cell begins and ends, is made of a double layer of special lipids called phospholipids. Because they repel water, they create a stable barrier that prevents the contents of the cell from dissolving away. Without lipids, there would be no cells, only a soup of chemicals. But lipids are also energy. They are the most concentrated form of energy storage in the body. A gram of fat holds more than twice the energy of a gram of carbohydrate. They are the deep reserves, the insulation that keeps you warm, and the cushion that protects your vital organs. Now, we arrive at the most complex and versatile of all biological molecules, the proteins. If carbohydrates are the fuel and lipids are the walls, proteins are the workers. They are the machines that actually do things. Proteins are made of long chains of smaller units called amino acids. There are 20 different standard amino acids used by life. Think of them as an alphabet of 20 letters. Depending on how you arrange these letters, you can write an infinite number of words. The cell strings these amino acids together in a specific order, and then in a feat of molecular origami, the chain folds up into a precise 3-dimensional shape. Form dictates function. This is a mantra of biology. The shape of the protein determines what it does. Some proteins form long, strong fibers, like the keratin in your hair or the collagen in your skin. These are structural proteins. Others fold into compact, globular shapes to become enzymes. Enzymes are the catalysts of life. They grab onto other molecules and speed up chemical reactions. Every time you digest food, contract a muscle, or sense light with your eyes, it is a protein enzyme making it happen. Hemoglobin is a protein in your blood, shaped perfectly to hold onto oxygen. Antibodies are proteins shaped perfectly to grab onto viruses and bacteria. The diversity of proteins is staggering. A single cell may contain thousands of different types, each performing a unique task to keep you alive. Finally, we reach the 4th family, the nucleic acids. These are the master planners. They do not build structures or burn for energy. Their job is information. They carry the blueprints for life. There are two main types: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. These are polymers made of monomers called nucleotides. We will explore the structure of DNA in great depth in a later lesson, but for now, understand its purpose. DNA resides in the nucleus, safe and protected. It holds the instructions for building every single protein in your body. It is the library. RNA is the messenger. It takes copies of those instructions out to the construction sites where proteins are built. Without nucleic acids, the cell would not know what to do. It would have the bricks, lipids and carbohydrates, and the workers, proteins, but no architectural plan. The nucleic acids ensure that a heart cell builds the proteins needed to beat, and a skin cell builds the proteins needed to protect. They ensure continuity, passing the instructions from one generation of cells to the next. Let us synthesize this view of the cell. Imagine the cell as a busy factory floor. The walls of the factory and the smaller rooms inside are built of lipids, creating waterproof barriers. The power generators are burning carbohydrates to keep the lights on and the machinery running. The machines themselves, the conveyor belts, the robotic arms, the quality control scanners, are the proteins, working tirelessly. And in the manager's office, the blueprints and hard drives storing all the operational data are the nucleic acids. All of these components interact in a watery gel called the cytosol. Water is the solvent of life. It surrounds these macromolecules, allowing them to float, collide, and react. The polarity of water molecules helps shape the proteins and membranes. It is the medium in which the chemistry of life takes place. As you drift deeper into relaxation, picture these molecules. They are not static. They are vibrating with thermal energy. They are constantly assembling and disassembling. This is your metabolism. Right now, in your sleep, your body is breaking down old proteins and building new ones. It is reading the nucleic acid blueprints to repair the wear and tear of the day. It is burning lipids and carbohydrates to maintain your body temperature. You are a masterpiece of chemistry. You are a collection of atoms, carbon, hydrogen, oxygen, nitrogen, organized into structures of immense complexity. From the chaos of the universe, life has ordered these elements into the cohesive, functioning miracle that is you. Let this knowledge comfort you. The machinery is sound. The chemistry is balanced. You do not need to direct it. It happens automatically, guided by billions of years of refinement. You can trust your cells to do their work while you rest. Transition your mind now from the individual molecules to the larger structure they create. All of these components, the lipids, the proteins, the sugars, come together to form the outer boundary of the living unit. Our next lesson will explore this dynamic barrier. We will learn how the cell defines itself against the world. Lesson 3. The cell membrane structure and function. Let your breath match the rhythm of your mind. Slow, steady, deep. With every inhale, imagine you are drawing in peace and clarity. With every exhale, release the tension of the physical world. You are floating in a space of pure understanding. We have explored the cell as a concept, and we have examined the chemical clay from which it is molded. Now, we must look at the architecture. Specifically, we must look at the wall. Every city needs a border. Every house needs a door. Without a boundary, the internal order of the cell would simply dissolve into the external chaos of the universe. This boundary is the cell membrane, also known as the plasma membrane. It is the skin of the cell. It defines where you begin and where the rest of the world ends. But this is no ordinary wall. It is not made of stone or brick. It is a dynamic, living skin, a gatekeeper that decides, moment by moment, what enters the sanctuary of the cell and what must be kept out. To understand this barrier, we must zoom in to the molecular level, back to the lipids we discussed in our last lesson. The foundation of the membrane is a special molecule called a phospholipid. Picture it clearly in your mind. It looks a bit like a balloon with two strings. The balloon part is the phosphate head. It is hydrophilic. It loves water. It wants to be near the watery fluids that fill your body. The strings are fatty acid tails. They are hydrophobic. They fear water. They want to get away from it. Now, imagine dropping millions of these molecules into water. A miraculous choreography occurs. To protect their tails from the water, they self-assemble into a double layer. The heads face outward towards the water on the outside and the water on the inside of the cell. The tails tuck themselves inside the sandwich, hiding away, creating a dry, oily core. This is the phospholipid bilayer. It is a soft, flexible film, only 2 molecules thick, yet it forms the boundary of every living cell on Earth. But a simple wall of fat would be too isolating. The cell needs to eat, to breathe, to talk, and so this bilayer is studded with other molecules. Scientists call this the fluid mosaic model. Imagine the membrane as a vast, calm sea. The phospholipids are the water, moving and flowing. Floating in this sea, like icebergs or boats, are proteins. This is the mosaic. The membrane is not a static sheet, it is a fluid, moving surface. The proteins float within it, sometimes drifting freely, sometimes anchored in place like buoys. These proteins are the workers of the wall. Some are integral proteins, buried deep within the membrane, spanning from one side to the other. These act as tunnels or channels. Others are peripheral proteins, sitting loosely on the surface, acting as anchors or identification tags. There is also cholesterol tucked between the tails of the lipids. In the human body, cholesterol often gets a bad reputation, but here it is a hero. It acts as a temperature buffer. When it gets cold, cholesterol keeps the tails from packing too tightly and freezing. When it gets hot, it stops them from becoming too fluid and melting. It maintains the perfect texture of the membrane, fluid enough to move, but solid enough to protect. So, what is the function of this complex skin? Its primary job is selective permeability. The membrane is a bouncer at the club. It is strictly selective about who gets in. Because the core of the membrane is oily and hydrophobic, small, non-polar molecules can slip right through. Oxygen, which you need to breathe, and carbon dioxide, which you need to exhale, drift through the membrane walls like ghosts walking through walls. They need no help. This movement is called simple diffusion. It is passive. It requires no energy. It is simply the nature of things to move from crowded areas to empty areas, from high concentration to low concentration. Imagine a drop of dye dispersing in a glass of water. The oxygen spreads into the cell in the same way. But what about the things the cell needs that are large or charged, like sugar or ions? They cannot pass through the oily core. They would be repelled. For these, the cell opens its doors. This is facilitated diffusion. The integral proteins act as specialized channels. Imagine a tunnel specifically sized and shaped for a glucose molecule. The sugar enters the tunnel and slides into the cell, still moving down its concentration gradient, still requiring no energy, but guided by the protein helper. However, sometimes the cell needs to hoard resources. Suppose the cell already has a lot of sugar, but it wants more. It needs to pull sugar in against the natural flow, like pumping water uphill. This requires work. This requires energy. This is active transport. The proteins here act not as open tunnels, but as pumps. The most famous of these is the sodium-potassium pump. It is working in your body right now, in every nerve and muscle. It uses ATP, the cell's energy currency, to grab sodium ions from inside the cell and force them out, while grabbing potassium ions from outside and dragging them in. Pump, pump, pump. It never stops. This constant pumping creates an electrical charge across the membrane. It effectively turns the cell into a tiny battery. When you think a thought or move your finger, you are discharging this battery. That spark of life is created by the work of the membrane pumps. Finally, for truly large items like a white blood cell trying to eat a bacteria, the membrane does something extraordinary. It is flexible, remember? It can fold inward, wrapping around the object until it swallows it whole, pinching off to form a bubble inside the cell. This is endocytosis. Conversely, the cell can package waste into a bubble, fuse it with the membrane, and spit it out. This is exocytosis. Think of the real-world implications of this barrier. When a person has diabetes, specifically type 2, the problem is at the membrane. The insulin signal arrives at the door, but the cell refuses to open the glucose channels. The sugar stays in the blood, unable to cross the barrier where it is needed. When a person has cystic fibrosis, a specific channel protein that moves chloride ions is misshapen. It's a broken door. The result is a buildup of mucus, all because one tiny protein in the mosaic is not doing its job. The cell membrane is a place of constant, frenetic activity, yet it maintains a stable, serene interior. It creates a self, distinct from the other. It is the first requirement for life. Without it, the chemicals of life would drift away into the ocean. With it, they are held together, allowed to interact, allowed to evolve. As you drift closer to sleep, visualize this barrier surrounding you. Not a hard shell, but a soft, shimmering bubble of protection. It allows the good to enter the oxygen, the nutrients, the rest. It keeps the harmful out. It pumps out the stress, the noise, the tension. You are safe within the membrane of your own rest. We have stood at the gates. We have examined the walls. Now, the gates are opening. We are invited inside. The interior of the cell is not empty space. It is a bustling metropolis of microscopic machinery. Our next lesson will take us on a tour of the internal structures that keep the cell alive. Lesson 4. The Cytoplasm and Organelles. Welcome to the inner sanctum of life. Welcome to the cytoplasm. If you were to shrink down and float inside a living eukaryotic cell, the first thing you would notice is that it is not empty space. You are not floating in a vacuum, nor are you in simple water. You are immersed in a thick, semi-transparent jelly called the cytosol. This fluid is the medium of life. It is rich with dissolved nutrients, ions, and enzymes. It provides the cushion and the suspension for everything else we are about to see. The term cytoplasm refers to everything within the cell membrane, excluding the nucleus. It is the landscape of the cell, a busy, crowded, and vibrant environment where the chemistry of existence takes place. Floating within this gel are the organelles. The word literally means little organs. Just as your body has a heart, lungs, and a liver, the cell has its own specialized structures, each enclosed in its own membrane, each performing a vital task to keep the whole system alive. We will tour these structures now, moving from the center outward. Dominating the center of this cellular city is the nucleus. It is the largest and most prominent organelle, often visible even with simple microscopes. Imagine a great, spherical vault. This is the command center. It houses the cell's genetic library, the DNA. The nucleus is surrounded by a double membrane called the nuclear envelope. This is not a solid wall, but a highly regulated barrier, punctuated by thousands of tiny openings called nuclear pores. These pores are the gatekeepers of the library, allowing specific molecules to enter and messenger RNA to exit, while keeping the precious DNA safely locked inside. Deep within the nucleus, you might see a darker, denser region. This is the nucleolus. It is a factory within a factory. Here, the cell constructs the parts for its most fundamental machines, the ribosomes. It is a place of intense activity, synthesizing ribosomal RNA and combining it with proteins. Let us move out through a nuclear pore and back into the cytoplasm. As we emerge, we find ourselves in a maze. This is the endoplasmic reticulum, or ER for short. The name sounds complex, but break it down. Endoplasmic means inside the cytoplasm, and reticulum means network. It is literally a network within the fluid. The ER is a vast system of folded membrane sacs and channels that are physically connected to the nuclear envelope. It winds its way through the cell like a series of conveyor belts. There are two distinct types of ER, defined by their appearance and their function. First, closest to the nucleus, is the rough endoplasmic reticulum. It is called rough because its surface is studded with millions of tiny granules. These granules are ribosomes. Ribosomes are the smallest organelles, but they are arguably the most important. They are the workers we mentioned in Lesson 2. They are not membrane-bound. They are complex molecular machines made of RNA and protein. Their job is to read the genetic instructions sent from the nucleus and build proteins. Imagine the rough ER as a busy manufacturing floor. The ribosomes act as the assembly workers, stitching amino acids together into long chains. As these chains are built, they are fed directly into the hollow space inside the ER, where they begin to fold into their correct three-dimensional shapes. The rough ER is the birthplace of proteins that are destined to be shipped out of the cell or embedded in the cell membrane. Further out, the network changes, the ribosomes disappear, and the membrane becomes tubular and smooth. This is the smooth endoplasmic reticulum. Since it lacks ribosomes, it does not make proteins. Instead, it acts as a specialized workshop for other tasks. The smooth ER is where the cell synthesizes lipids. It builds the phospholipids that repair the membrane and the steroid hormones that signal the body. It also acts as a detoxification center. In the cells of your liver, the smooth ER is massive, working tirelessly to break down alcohol, drugs, and metabolic wastes, making them safe for the body to excrete. It also stores calcium ions, which are crucial for triggering muscle contractions. It is a silent, smooth operator, handling the chemistry of fats and poisons. But what happens to the proteins and lipids after they are made in the ER? They cannot just float away. They need to be packaged and addressed. They need a post office. Small bubbles of membrane, called transport vesicles, pinch off from the ER, carrying their cargo of newly made proteins. They drift through the cytosol and arrive at our next stop, the Golgi apparatus. Named after the Italian scientist Camillo Golgi, who discovered it, this organelle looks like a stack of flattened pancakes or dinner plates, slightly curved. It is distinct from the ER. It has a receiving side, called the cis face, which faces the ER, and a shipping side, called the trans face, which points toward the cell membrane. The vesicles from the ER fuse with the Golgi's receiving side, dumping their protein cargo into the stack. As the proteins move through the layers of the Golgi, they undergo a transformation. Enzymes inside the Golgi modify them, adding a sugar tag here, trimming a molecular tail there. These modifications act like shipping labels or stamps. They tell the cell exactly where this protein needs to go. Finally, the proteins reach the shipping side. The membrane of the Golgi bulges out and pinches off, creating new secretory vesicles. These are the delivery trucks. Some are destined for the cell membrane, where they will fuse and release their contents to the outside world, perhaps sending insulin into the bloodstream or neurotransmitters into a synapse. Others are destined for other parts of the cell. One such destination is the lysosome. These are specialized vesicles produced by the Golgi that contain powerful digestive enzymes. If the nucleus is the library and the Golgi is the post office, the lysosome is the recycling center. The cell is a clean place. It does not tolerate waste. When an organelle becomes old and broken, or when the cell engulfs a piece of food, the lysosome fuses with it. The enzymes inside, which are so powerful they would dissolve the cell itself if they ever leaked out, break down the waste into raw materials. Proteins are chopped back into amino acids. Carbohydrates are broken back into simple sugars. These raw materials are then released back into the cytosol to be reused. It is a perfect cycle of renewal. Consider this connected system. The nucleus holds the plan. The ribosomes on the rough ER build the product. The smooth ER builds the packaging. The Golgi creates the label and ships the package. And the lysosome cleans up the scraps. Biologists call this the endomembrane system. It is a marvel of logistics, occurring billions of times a day within you. Now, pause and visualize this activity. It is not chaotic, it is rhythmic. Vesicles, bud and fuse, flowing like drops of oil in a lava lamp. The factory is humming with a quiet, efficient purpose. There are real-world consequences when this factory breaks down. Consider the genetic disorder Tay-Sachs disease. In this condition, a single enzyme in the lysosome is missing. The recycling center cannot break down a specific type of lipid found in brain cells. As a result, the lipid piles up. Trash accumulates in the cell until it swells and eventually dies. It is a tragedy caused by a microscopic failure in the organelle's machinery. It reminds us of how precise this system must be. But for you, right now, the system is working perfectly. Your cells are synthesizing exactly what you need. They are detoxifying your blood, They are recycling the old to make way for the new. The city is safe. The walls are strong. The workers are skilled. As we drift away from the endomembrane system, we notice other structures floating in the cytosol, structures that generate the power to run all these pumps and belts, structures that have their own ancient history. Our next lesson will take us into the power plants of the cell. We will explore the source of your body heat and the fuel of your movement. Lesson 5. Bioenergetics, Mitochondria and Chloroplasts. We have built the walls of the cell. We have toured the factory floor where proteins are made, but a factory cannot run without electricity. A city cannot function without a power grid. Life is a constant battle against entropy. It requires a relentless input of energy to maintain order, to move, to think, and to grow. Without energy, the complex structures we have visualized would simply collapse back into dust. In this lesson, we visit the power plants of the biological world. We will explore the organelles of bioenergetics, the mitochondria, and the chloroplasts. Let us begin with the mitochondrion. You may find this organelle in nearly every eukaryotic cell, from the neurons in your brain to the muscles in your legs. It is often called the powerhouse of the cell, but that phrase, while true, is too simple. It is more like a highly sophisticated refinery. Visualize its shape. It is roughly the size of a bacterium, often bean-shaped or oval. Unlike the other organelles we have seen, the mitochondrion is wrapped in not one, but two membranes. The outer membrane is smooth, acting as a skin to separate it from the cytoplasm. But the inner membrane is where the magic happens. It is highly folded, wrinkling back and forth into deep ridges called cristae. Why the folds? In biology, surface area is everything. These folds drastically increase the amount of space available for chemical reactions. It is like crumpling a large sheet of paper to fit it into a small cup. Inside this inner membrane lies a fluid-filled space called the matrix, which is packed with enzymes and DNA. The function of the mitochondrion is cellular respiration. This is the reason you breathe. The cell takes the glucose derived from your food and the oxygen you inhale and sends them to the mitochondrion. Inside this refinery, through a complex series of reactions known as the Krebs cycle and the electron transport chain, the energy locked in the bonds of the sugar is released. But it is not an explosion, it is a controlled burn. The mitochondrion carefully harvests this energy to charge a molecule called ATP, adenosine triphosphate. Think of ATP as a fully charged, rechargeable battery. It is the universal energy currency of life. Every time you twitch a muscle, your cells spend an ATP molecule, breaking it down to release the energy needed for the movement. The mitochondrion then takes the dead battery and recharges it, over and over again. A working muscle cell recycles its entire pool of ATP once every minute. That is the intensity of the work happening inside these silent, golden organelles. Now, let us shift our gaze to the other side of the bioenergetic coin. We move from the animal to the plant. We move from the consumer to the producer. We encounter the chloroplast. If the mitochondrion is a furnace burning fuel, the chloroplast is a solar panel capturing the sun. Found only in plants and algae, these organelles are the reason the world is green. Like the mitochondrion, the chloroplast has a double membrane, but inside, the structure is different. Floating within the inner fluid called the stroma are stacks of coin-shaped sacs called thylakoids. These stacks look like towers of green poker chips. Embedded in the membranes of these thylakoids is a pigment called chlorophyll. Chlorophyll is a miracle molecule. It has the unique ability to absorb the energy of sunlight. When a photon of light hits a molecule of chlorophyll, it excites an electron, kicking it to a higher energy state. This is the spark that starts the engine of photosynthesis. The chloroplast takes this light energy and uses it to split water molecules apart, releasing oxygen as a waste product. This is the oxygen we breathe. It then uses that energy to bind carbon dioxide from the air into solid sugar. It is alchemy. It turns light and air into matter. Consider the beautiful symmetry between these two organelles. The chloroplast builds the sugar and releases oxygen. The mitochondrion burns the sugar and consumes the oxygen, releasing carbon dioxide. The carbon dioxide returns to the chloroplast to be made into sugar again. It is a perfect, closed loop. The breath of the plant becomes the life of the animal. The waste of the animal becomes the food of the plant. But there is a deeper secret here, one that connects you to the ancient past. As scientists studied mitochondria and chloroplasts, they noticed something strange. These organelles look remarkably like bacteria. They are about the same size. They have their own circular DNA, distinct from the DNA in the nucleus. They reproduce on their own schedule, splitting in two, even when the cell isn't dividing. They have their own ribosomes, which look more like bacterial ribosomes than eukaryotic ones. This led to the endosymbiotic theory. Visualize the Earth billions of years ago, a world of single-celled organisms. There was a large, predatory cell, an ancestor of the eukaryote. One day, it engulfed a small, efficient, energy-producing bacterium. But instead of digesting it, the large cell kept it. The small bacterium provided extra energy in the form of ATP. The large cell provided protection and raw materials. Over millions of years, they became inseparable. The bacterium became the mitochondrion. Later, one of these cells engulfed a photosynthetic bacterium, which became the chloroplast. This means that you are not just one organism, you are a colony. Every cell in your body houses the descendants of those ancient bacteria. They are living guests, working within you, powering your existence. This relationship has real-world consequences for your health. Because mitochondria have their own DNA, they can have their own genetic mutations. These result in mitochondrial diseases, which often affect energy-hungry tissues like muscles and the brain. Interestingly, because sperm cells are small and contribute almost no cytoplasm to the egg, you inherit your mitochondria exclusively from your mother. If you traced your mitochondrial DNA back, you could follow a direct maternal line through your mother, and her mother, stretching back to the dawn of humanity. Think also of your stamina. When you exercise, when you run, swim, or lift weights, you are putting a demand on your mitochondria. If you train for endurance, your body responds not just by building bigger muscles, but by building more mitochondria within the muscle cells. You literally build more power plants to handle the load. That feeling of getting in shape is the feeling of increased mitochondrial efficiency. As you lie here now, completely still, your mitochondria are working. They are quietly burning fuel to keep your body temperature at a steady 98.6 degrees. They are charging the ATP batteries that your heart needs to beat. They are powering the neurons that are processing these words. Imagine a soft, golden warmth spreading through your body. This is the heat of cellular respiration. It is the fire of life, controlled, contained, and sustaining. You don't need to stoke the fire, it burns on its own. Now that we understand the power source, we must ask, what does the cell do with this energy? It builds, it moves, it maintains its shape. But a cell is not a shapeless balloon. It has a structure, it has a skeleton. Our next lesson will explore the internal scaffolding that holds the cell together and allows it to move through the world. We will learn about the beams, the ropes, and the tracks that crisscross the interior. Lesson 6. The Cytoskeleton and Cell Motility. Welcome to the world of the cytoskeleton. Unlike your own skeleton, which is made of hard, calcified bone, the cell's skeleton is dynamic. It is made of protein fibers that can grow, shrink, assemble, and disassemble in seconds. It is a scaffolding that is constantly rebuilding itself. To understand this network, we must look at its three distinct components, arranged by size from the thinnest to the thickest. First, let us examine the microfilaments. Imagine the finest, most delicate threads. These are made of a protein called actin. Actin filaments are solid rods, twisted like a double helix. They are most concentrated just beneath the cell membrane, forming a dense web called the cortex. This web gives the cell its shape and consistency. Think of the gel-like outer layer of an amoeba. That stiffness is provided by microfilaments. But actin is not just for structure, it is for movement. Actin works in partnership with another protein called myosin. You already know this partnership intimately. Every time you move a muscle, millions of myosin heads are grabbing onto actin filaments and pulling. This sliding action shortens the muscle fiber, creating contraction. But even in non-muscle cells, actin is active. When a white blood cell crawls through your tissue to chase down a bacteria, it is pushing its membrane forward by rapidly building actin filaments at the leading edge. It is literally constructing a road in front of itself to drive upon. Next, we move to the middle-sized fibers, the intermediate filaments. If actin is for movement, intermediate filaments are for strength. They are rope-like fibers, woven together like steel cables. They are built from a family of proteins that includes keratin, the same tough protein found in your hair and fingernails. The primary job of intermediate filaments is to bear tension. They stretch across the cell, anchoring the nucleus in place and connecting to the cell membrane at strong adhesion points. Imagine a suspension bridge. The cables that hold the bridge up against the wind and gravity are the intermediate filaments. Without them, the cell would rupture under stress. They are the reason your skin does not tear when you stretch it. They provide the mechanical integrity of your tissues. Finally, we arrive at the largest and most complex components, the microtubules. These are not solid rods, they are hollow tubes, like microscopic pipes. They are built from a protein called tubulin. Microtubules are stiff and rigid. They radiate outward from a central point near the nucleus called the centrosome. They act as the primary structural girders of the cell, resisting compression. If intermediate filaments are the tension cables, microtubules are the steel beams that keep the building from collapsing inward. But microtubules are more than just beams. They are the highways of the cell. Recall the factory we toured in Lesson 4. We saw vesicles carrying proteins from the ER to the Golgi, but how do they move? They do not simply float randomly. The cytoplasm is too crowded for that. Instead, they travel along the microtubule tracks. This brings us to one of the most fascinating mechanisms in all of biology, the motor proteins. Visualize a microtubule stretching across the dark space of the cell. Now, see a tiny protein molecule attaching to it. This is kinesin. It looks remarkably like a person. It has 2 feet that attach to the microtubule and a long tail that holds onto a cargo vesicle, which might be thousands of times its size. Using ATP for energy, the kinesin molecule literally walks. One foot swings forward and binds, then the other foot swings forward, step by step by step. It marches along the microtubule highway, carrying its precious cargo to the edge of the cell. Another motor protein, Di9, walks in the opposite direction, carrying waste back toward the center. This is happening inside you right now, in the long axons of your nerve cells, which can stretch from your spine to your toes. Kinesin motors are marching for days to deliver neurotransmitters to the nerve endings. It is a bustling, organized logistical network, operating on a scale so small it defies imagination. But the cytoskeleton is capable of even grander movements. It is the engine of cell motility. Some cells need to swim. A sperm cell, for example, must travel a great distance to reach the egg. To do this, it uses a flagellum. This is a long, whip-like tail. If you were to slice a flagellum open, you would see a beautiful geometric arrangement of microtubules inside 9 pairs in a ring, with 2 in the center. Motor proteins between these pairs pull and release, causing the microtubules to slide past each other. This sliding motion causes the entire tail to bend and snap back, propelling the cell forward. Other cells, like the ones lining your windpipe, have thousands of short, hair-like projections called cilia. These also contain microtubules. They beat in a rhythmic, coordinated wave, like a field of wheat in the wind. This beating motion pushes mucus and trapped dust out of your lungs, keeping your airways clear. The cytoskeleton is also the master architect of cell division. When a cell decides to divide, the entire microtubule network disassembles and rebuilds itself into a structure called the mitotic spindle. This spindle grabs onto the chromosomes and physically pulls them apart, ensuring that each new daughter cell gets a perfect copy of the genetic code. It is a delicate, high-stakes ballet orchestrated by tubulin and motors. Consider the real-world implications of this system. Because microtubules are so vital for cell division, they are a primary target for cancer treatments. Drugs like Taxol work by freezing the microtubules in place. If the microtubules cannot disassemble and move, the cell cannot divide. The cancer cell, unable to replicate, dies. We use the cell's own skeleton against it to stop the disease. However, defects in the cytoskeleton can also cause disease. In epidermolysis bullosa, a genetic condition, the intermediate filaments in the skin are defective. Without these reinforcing cables, the skin becomes as fragile as a butterfly's wing, blistering at the slightest touch. It reminds us that our strength, our literal physical toughness, begins at the molecular level. As you visualize these networks, the twisting actin, the ropey keratin, the hollow tubulin, understand that they are never still. The cytoskeleton is in a constant state of flux, responding to the needs of the cell. If a cell needs to move left, it builds skeleton to the left. If it needs to divide, it rebuilds itself entirely. It is a structure of infinite adaptability. Let this image support you. You are built of trillions of these tiny, resilient structures. They hold you together. They pull you forward. They are the unseen strength that allows you to stand, to move, to be. Feel the tension in your own body releasing. Just as the cytoskeleton can disassemble to let the cell rest, you can let your own muscles soften. You do not need to hold yourself up right now. The bed is your support. The world is your support. We have explored the body of the cell, its organs, its energy, and its bones. But a body is nothing without connection. A cell does not exist in a void. It must anchor itself to its neighbors. It must join a community to form a tissue. Our next lesson will explore how cells hold hands. We will look at the rivets, the glues, and the channels that bind us together. Lesson 7. Cell junctions and the extracellular matrix. In this lesson, we explore the architecture of connection. We will examine how cells bind themselves together to form tissues, and we will discover the complex environment in which they live. We leave the interior and step into the space between. This is the world of cell junctions and the extracellular matrix. Imagine you are standing in a dense forest. The trees are the cells. They are distinct individuals, yet they are rooted in a common soil, their branches interlaced, creating a canopy that acts as one living entity. In the body, this soil is the extracellular matrix, or ECM. The ECM is not empty space. It is a dense physical meshwork that provides structural support to the cells. It is built primarily of glycoproteins, proteins with carbohydrate chains attached. The most abundant of these is collagen. Visualize collagen. It is the steel of the biological world. It forms thick, ropey fibers that are incredibly strong. In fact, gram for gram, collagen is stronger than steel. It weaves through the space between your cells, creating a trellis that resists stretching. This is what gives your skin its toughness. When you pinch the back of your hand and it snaps back into place, you are feeling the resilience of the collagen matrix. It anchors the cells, giving them something to grab onto. Interwoven with the collagen are other proteins, like fibronectin, which acts as a sticky glue, and proteoglycans. These proteoglycans are like molecular sponges. They trap water, turning the matrix into a hydrated gel. This gel cushions the cells, protecting them from compression and allowing nutrients to diffuse easily from the blood. But the cell does not just float passively in this mesh. It connects to it. Spanning the cell membrane are special receptor proteins called integrins. Imagine these as grappling hooks. On the inside of the cell, the integrin grabs onto the cytoskeleton, the actin filaments we discussed in our last lesson. On the outside, it latches onto the collagen of the ECM. This connection is profound. It means that the inside of the cell is physically linked to the outside world. If the matrix stretches, the cell feels it. If the matrix stiffens, the cell knows. This mechanical feedback tells the cell where it is and what it should be doing. It is how a stem cell knows to become a bone cell or a muscle cell by feeling the stiffness of the bed it lies in. Now, let us look at how cells connect directly to one another. In multicellular organisms like us, cells are rarely solitary. They pack together to form sheets and tubes, the lining of your gut, the surface of your skin, the walls of your blood vessels. To maintain the integrity of these barriers, cells use specialized structures called cell junctions. There are three main types you should visualize. First, Imagine a watertight seal. This is the tight junction. Picture two cells pressing their membranes together. Proteins stitch them so tightly that there is virtually no space between them. It is like a Ziploc bag or the seams of a waterproof coat. You find these junctions in places where leakage would be dangerous. Consider your stomach. It is filled with strong acid. If that acid were to leak between the cells of your stomach lining, it would digest your own body. Tight junctions prevent this. They ensure that fluids must go through the cell, where they can be regulated, rather than slipping between them. They create a distinct boundary between the chaotic external world and the controlled internal body. Second, imagine a spot weld or a rivet. This is the desmosome. Sometimes, cells need to be held together, but they also need to be flexible. They need to endure stress and stretching. Tight junctions are too brittle for this. They might tear. So, the body uses desmosomes. These are button-like points of adhesion. Inside the cell, they are anchored to the intermediate filaments, the tough keratin cables we learned about. When two cells are riveted together by desmosomes, and those rivets are anchored to the internal skeletons, the stress is distributed across the entire sheet of tissue. This is crucial in tissues like your skin and your heart muscle. Every time your heart beats, the cells pull against each other. Without desmosomes, your heart cells would rip apart under the strain. They allow the tissue to act as a single, mechanical sheet, flexing and beating without breaking. Third, imagine a tunnel. This is the gap junction. So far, we have discussed structural connections, but cells also need to communicate. They need to share. Gap junctions are not about strength. They are about flow. They are formed by ring-like proteins that create open channels directly connecting the cytoplasm of one cell to the cytoplasm of its neighbor. These channels are tiny, too small for proteins or DNA to pass through, but they are large enough for ions, sugars, and small signaling molecules. Through these gaps, cells can share food. They can share chemical distress signals. Most importantly, they can share electricity. Think of your heart again. For your heart to be an effective pump, thousands of muscle cells must contract at the exact same instant. They cannot wait for a chemical messenger to swim from one cell to the next. That would be too slow. Instead, the electrical signal flows instantly through the gap junctions, washing over the heart muscle like a wave. It allows the tissue to behave as if it were one giant cell. This is unity in action. There is a fourth type of junction, found only in plants, called the plasmodesmata. Since plant cells are trapped inside thick, rigid cell walls, they cannot touch directly. Instead, they have plasmodesmata channels that punch through the cell walls, connecting the living interiors of adjacent plant cells. It turns the entire plant into a continuous living network, allowing water and nutrients to flow from the roots to the leaves without ever crossing a membrane. Why does this matter to you resting here right now? It matters because you are not a pile of cells. You are a unified organism. The breakdown of these connections is the root of many diseases. When cancer cells become malignant, one of the first things they do is destroy their desmosomes and detach from the extracellular matrix. They cut their tethers. This allows them to float away, to metastasize, and spread to other parts of the body. A cancer cell is a cell that has forgotten its connection to the community. Conversely, the aging process is largely a story of the extracellular matrix. As we get older, the collagen in our skin breaks down and becomes disorganized. The elastic fibers lose their snap, the soil becomes loose, and the skin begins to wrinkle and sag. It is a reminder that our structure depends not just on the cells themselves, but on the strength of the web that holds them. But tonight, your matrix is strong. Your collagen is holding you together. Your tight junctions are protecting your organs. Your gap junctions are allowing your heart to beat in a slow, rhythmic lullaby. Visualize this web of connection. See the trillions of cells in your body holding tight to one another. There is safety in this connection. No cell is alone. They support each other. They feed each other. They talk to each other. You are a vast, cooperative community. You are a galaxy of connected stars. Let this feeling of unity wash over you. The tension in your muscles is melting away because the cells know they are supported. They do not need to fight gravity alone. We have established the physical connections. the stage is set. But a community needs more than just physical proximity. It needs a language. It needs a way to send complex orders, to warn of danger, to coordinate growth. Our next lesson will explore the whispers and shouts of the cellular world. We will learn how a chemical released in your brain can tell a cell in your toe to wiggle. Lesson 8. Cell Signaling and Communication. In this lesson, we explore the invisible language of biology. We delve into cell signaling and communication. Imagine you are standing in a crowded room. To get someone's attention, you might wave your hand, or you might shout their name. Cells do not have hands to wave or voices to shout. Instead, they use chemistry. They speak in molecules. The words of their language are specific chemical compounds called ligands. These can be anything from small gas molecules to large proteins, hormones, or neurotransmitters. But a word is useless if no one is listening. For a cell to hear, it must have ears. In the cellular world, these ears are receptor proteins. We discussed these briefly when we looked at the membrane. A receptor is a protein that sits on the surface of the cell, or sometimes inside it, waiting for a specific message. It is highly specific. A receptor for insulin will only listen to insulin. It will ignore adrenaline, dopamine, or estrogen. It is like a lock that can only be opened by one specific key. The process of communication happens in 3 distinct stages: reception, transduction, and response. Let us walk through this journey, step by step. The first stage is reception. Visualize a single signaling molecule, let's say, a molecule of adrenaline floating through the bloodstream. It approaches a cell. It bumps into the membrane. It finds its matching receptor. The moment the ligand binds to the receptor, something magical happens. The receptor changes shape. Just as a door handle turns when you insert the key, the protein shifts its physical form. This shape change is the signal. It tells the inside of the cell, someone is knocking. There is news from the outside. The second stage is transduction. This is where the signal is relayed. The receptor does not usually perform the final task itself. Instead, it passes the message along like a baton in a relay race. The changed receptor activates a protein inside the membrane, which activates another, which activates another. This is called a signal transduction pathway. Why this complexity? Why not just have the receptor do the job? The answer is amplification. Imagine a single general giving an order to 2 colonels. Each colonel gives the order to 10 captains. Each captain gives the order to 100 soldiers. A single whisper from the general results in a roar from the army. Similarly, one single molecule of adrenaline binding to the outside of the cell can trigger the release of millions of glucose molecules inside the cell. The signal grows louder and stronger with every step of the relay. This relay often involves small, mobile molecules called second messengers. The most famous of these is cyclic AMP, or CAMP. When the first messenger, the hormone, hits the membrane, it triggers an enzyme to make thousands of CAMP molecules inside the cell. These little messengers dash through the cytoplasm, waking up other proteins, spreading the news instantly throughout the cellular city. Another common second messenger is the calcium ion. Usually, the cell works hard to keep calcium levels low in the cytoplasm, storing it away in the smooth endoplasmic reticulum. But when a signal arrives, the cell opens the floodgates. Calcium rushes out, and this sudden flood triggers immediate action, like the contraction of a muscle or the release of a neurotransmitter. Getty images the third and final stage is response. The message has arrived. The command has been amplified. Now, the cell must act. The response depends entirely on the cell's job. If it is a liver cell receiving a signal from insulin, the response might be to open channels and absorb sugar from the blood. If it is a skin cell receiving a signal from a growth factor, the response might be to enter the cell cycle and divide. If it is a cell with damaged DNA, the signal might be a command to self-destruct the process of apoptosis we will discuss later. Sometimes, the response happens in the cytoplasm, turning an enzyme on or off. Other times, the signal travels all the way into the nucleus, finding the DNA itself and turning specific genes on or off. This changes the very nature of what the cell is building. Let us ground this in a real-world example that you have likely experienced. Think of the fight or flight response. You are walking down a street and a dog barks aggressively at you. Instantly, before you even have time to think, "I am scared," your body reacts. This is cell signaling at the speed of survival. Your brain sends a signal to your adrenal glands. They release the ligand, adrenaline. The adrenaline travels through your blood to your liver cells. The receptors there catch the signal, trigger a transduction pathway, and the liver floods your blood with glucose for energy. Simultaneously, the adrenaline hits your heart cells. The receptors there trigger a different pathway, causing the heart muscle to contract faster and harder. It travels to the cells of your lungs, causing the airways to dilate so you can breathe more oxygen. It travels to the muscles of your digestive system and tells them to shut down. Digestion is not a priority right now. One single chemical word, adrenaline, is heard by different cells in different ways, coordinating a massive, full-body response to keep you safe. But what happens when the conversation goes wrong? Many diseases are essentially failures of communication. Consider type 2 diabetes. The pancreas is shouting, "Insulin, insulin," meaning, "take up sugar," but the cells have become deaf. Their receptors have stopped responding effectively. The signal is lost, the sugar remains in the blood, and the body suffers. Consider autoimmune diseases. Here, the immune system confuses the signal for friend with the signal for enemy, attacking the body's own tissues. Consider cancer. Cancer cells often have mutations in their signaling pathways. They might have a broken receptor that is permanently stuck in the on position, telling the cell, "Grow, grow, grow," even when there is no growth signal present. It is like a doorbell that keeps ringing even when no one is pressing the button. There is also a deeper, quieter form of communication called quorum sensing. This was discovered in bacteria, but our cells use versions of it too. Bacteria release small signaling molecules into their environment. When there are only a few bacteria, the concentration of the signal is low and nothing happens. But as the bacteria multiply and become crowded, the concentration of the signal rises. Once it hits a certain threshold, all the bacteria detect it simultaneously. They realize, "We are many." And then, in unison, they change their behavior. They might launch an attack or build a protective biofilm. It is democracy at the molecular level. As you lie here, your own cells are engaging in this constant, silent dialogue. Your brain is sending signals to your muscles to relax. Your stomach is signaling your brain that it is full or empty. Your immune cells are patrolling, flashing identity cards at one another, checking for intruders. It is a symphony of whispers. It is a ceaseless flow of information that keeps the chaos of the universe at bay. You do not need to direct this conversation. You do not need to tell your heart to listen to the adrenaline or your liver to listen to the insulin. It is encoded in the deep wisdom of your biology. The listeners are awake so that you can sleep. The sentries are watching so that you can rest. Visualize these signals as gentle streams of light flowing through your body, connecting the head to the heart to the hands. A network of light, a network of knowing. There is peace in this connection. The body is not a collection of strangers. It is a family that never stops talking to one another. We have explored the language. But where is the dictionary? Where are the definitions for all these proteins, receptors, and signals stored? They are locked away in the vault of the nucleus. Our next lesson will take us to the source code. We will open the library of life and examine the most famous molecule in history. Lesson 9. The structure of DNA. In every eukaryotic cell, there is a vault called the nucleus, and inside that vault lies the most famous molecule in the known universe. It is the master copy. It is the instruction manual for building you. In this lesson, we will explore the architecture of this molecule. We will learn how a simple chemical structure can hold the complex secrets of life. We will explore the structure of DNA. DNA stands for deoxyribonucleic acid. It is a long, elegant word for a long, elegant molecule. To understand it, we must act as architects. We must build it up from its smallest components. DNA is a polymer, which means it is a chain made of repeating links. These links are called nucleotides. Visualize a single nucleotide. It is made of 3 distinct parts. First, There is a sugar. This is not the sugar you put in your coffee. It is a specific 5-carbon sugar called deoxyribose. Imagine this sugar as a small, sturdy pentagon. It is the anchor. Attached to one side of the sugar is a phosphate group. This is a small cluster of phosphorus and oxygen atoms. It is negatively charged. If the sugar is the anchor, the phosphate is the hook that connects it to the next link in the chain. Attached to the other side of the sugar is the third and most important part, the nitrogenous base. This is the letter of the alphabet. In the language of DNA, there are only 4 letters, 4 bases. There is adenine, which we call A. There is thymine, which we call T. There is cytosine, which we call C. And there is guanine, which we call G. These 4 distinct shapes are the only vocabulary life uses. Whether you are a human, a bacteria, a mushroom, or a blue whale, your code is written in A, T, C, and G. Now, let us assemble the chain. The phosphate of one nucleotide connects to the sugar of the next. Sugar, phosphate, sugar, phosphate, sugar, phosphate. This creates a strong, repeating backbone. Imagine a long ribbon. The sugars and phosphates form the fabric of the ribbon, and the nitrogenous bases stick out from the side like teeth on a zipper. But DNA does not exist as a single ribbon. It is a double molecule. This was the great secret discovered in 1953 by James Watson and Francis Crick, using the critical X-ray images produced by Rosalind Franklin. They realized that DNA is formed by 2 strands winding around each other. Visualize this now. 2 long ribbons floating in the darkness. They come together. But they do not just touch randomly. They follow a strict, unbreakable rule called complementary base pairing. The bases on one strand reach out to touch the bases on the other strand, but they are picky. Adenine A will only ever hold hands with thymine T. They fit together like 2 puzzle pieces. Cytosine C will only ever hold hands with guanine G. This is the golden rule of biology. A pairs with T, C pairs with G. Why is this so important? It means that the 2 strands are perfect mirror images of each other. If you know the sequence of one strand, say AGTC, you automatically know the sequence of the other strand, TCAG. This complementary nature is the secret to how life copies itself. One strand acts as the template for the other. As these bases pair up in the center, they form the rungs of a ladder. The sugar-phosphate backbones form the side rails. But this ladder is not straight. The geometry of the bonds causes the entire structure to twist. It spirals clockwise, turning and turning. This is the double helix. It is a shape of immense beauty. It is robust, yet flexible. The bases, the precious code, are tucked safely in the middle of the spiral, protected from the chemical chaos of the cell by the sturdy sugar-phosphate rails on the outside. Consider the length of this molecule. If you were to take the DNA from a single human cell and stretch it out in a straight line, it would be about 2 meters long, 6 feet of code, coiled inside a nucleus that is microscopic. How is this possible? The DNA is not just stuffed in like spaghetti. It is organized. It is wrapped around spool-like proteins called histones. These spools are packed together into thicker fibers, which are looped and coiled again. This packaging is so efficient that the 6 feet of DNA fits into a space smaller than the tip of a needle. When the cell is preparing to divide, these coils tighten even further, becoming the dense, X-shaped structures we call chromosomes. But tonight, let us focus on the code itself. Think of the DNA as a library. The nucleus is the building. The chromosomes are the bookshelves. The genes are the books. A gene is simply a specific stretch of DNA that holds the instructions for building one specific protein. You might have a gene for blue eyes. That is a sequence of A's, T's, C's, and G's that tells the cell how to make a pigment protein for your iris. You have a gene for hemoglobin. That is a different sequence that tells the blood cells how to build the oxygen-carrying machine. You have roughly 20,000 of these gene books in your library. Most of your DNA, interestingly, is not genes. A huge portion of it is what we used to call junk DNA, but now realize is regulatory. These are the spaces between the books, the instructions that tell the cell when to read a book or how loudly to read it. The stability of this structure is crucial. You want your library to last. You don't want the letters changing on their own. The chemical bonds that hold the backbone together, covalent bonds, are very strong. They are hard to break. But the bonds that hold the two strands together in the middle, hydrogen bonds, are relatively weak. They are like a magnetic clasp. They are strong enough to hold the helix together, but weak enough that the cell can unzip the strands when it needs to read the code or copy it. This dual nature, strong lengthwise, unzippable widthwise, is the genius of the design. Reflect on the universality of this molecule. The DNA in your body uses the exact same chemistry as the DNA in an apple tree or a virus. We are all written in the same language, just telling different stories. It confirms that all life on Earth shares a single common ancestor. We are all branches of the same family tree, rooted in this double spiral. As you rest here, your cells are guarding this code. They have armies of enzymes constantly patrolling the DNA strands, looking for damage. If a T is accidentally knocked out by UV light from the sun, repair enzymes rush in, see the A on the other side, and know exactly what to put back. They stitch the T back in place. Your body is constantly proofreading itself, ensuring that the manual remains pristine. There is a deep comfort in this. You do not need to remember how to beat your heart. You do not need to remember how to digest your food or how to fight an infection. It is written down. It is safe. The wisdom of millions of years of evolution is coiled tightly within you, accessible whenever you need it. Visualize the spiral again. See it twisting slowly in the golden light. It is the thread of continuity. It connects you to your parents and to their parents, and back through the generations. It connects you to the future. It is the physical substance of history and potential. The code is beautiful. The code is secure. But a library is useless if the books are never opened. A manual is useless if it is never copied. For life to continue, this long, delicate molecule must be duplicated. One cell must become two. Our next lesson will explain how this incredible feat is accomplished. We will watch the helix unzip. We will watch the machinery of life build a perfect mirror image. Lesson 10. DNA. Replication. Before a cell can divide, before one life becomes two, it must duplicate its entire library. It cannot give half its books to the new cell. That would be a disaster. It must give a complete, perfect copy. This process is called DNA replication. It is happening in your body right now, in the cells of your skin, your gut, your bone marrow. It is an event of staggering speed and precision. Let us visualize this process as a coordinated dance of molecular machines. The DNA helix is stable. It likes being twisted shut. To copy it, we must first open it. The process begins at specific locations on the chromosome, called origins of replication. Imagine a zipper that is stuck closed. You need a tool to force it open. Enter the first machine, helicase. Visualize helicase as a spinning wedge, a motor that races down the DNA strand. Its job is to break the hydrogen bonds holding the base pairs together. It unzips the helix, separating the two strands. As it moves, it creates a replication fork, a Y-shaped junction where the single road splits into two. But DNA is under tension. Have you ever tried to pull apart two strands of a tightly twisted rope? The rope knots up and tangles further down the line. To prevent this, another enzyme called topoisomerase moves ahead of the fork. It snips the DNA, lets it untwist to release the tension, and then glues it back together. It clears the path for the helicase. Now the strands are exposed. They are naked and vulnerable. Single-strand binding proteins rush in to coat the DNA, keeping the two sides from snapping back together like magnets. The template is ready. Now comes the master builder, the star of the show, DNA polymerase. This is an enzyme shaped somewhat like a hand. It grips the DNA strand. Its job is to read the exposed letter on the old strand and grab the matching free-floating nucleotide from the soup of the nucleus to build the new strand. If it sees an adenine, it grabs a thymine and snaps it into place. If it sees a cytosine, it grabs a guanine. Click, click, click. It travels down the line, adding bases at a rate of about 50 per second in human cells. In bacteria, it can go 10 times faster. But there is a catch, a quirk of evolution. DNA polymerase is a bit like a train that can only run on tracks that already exist. It cannot start from nothing. It needs a starting block. So, another enzyme called primase comes in first. It lays down a short little stretch of RNA, not DNA, called a primer. This primer acts as the handle. DNA polymerase grabs the handle and begins ''its work. And here lies the great complication of replication. DNA strands have direction. We call the ends 5' and 3' based on the carbon numbering of the sugar. The two strands of the helix run in opposite directions. They are anti-parallel, like a two-way street. DNA polymerase is a one-way engine. It can only build in the 5 to 3 direction. This means that on one of the strands, the engine is moving in the same direction as the zipper. As helicase opens the fork, the polymerase follows right behind it, building one long, continuous strand. This is called the leading strand. It is smooth. It is easy. But on the other strand, the engine is facing the wrong way. It is moving away from the zipper. This is the lagging strand. Here, the cell has to work backwards. As the helicase opens a section, the primase puts down a primer, and the polymerase works backward to fill the gap. Then the helicase opens a bit more, and the polymerase has to jump back to the fork and work backward again. It is a 2 steps forward, 1 step back dance. This produces the new DNA in short, disconnected chunks. We call these Okazaki fragments, named after the husband and wife team Reiji and Tsuneko Okazaki who discovered them. Visualize this chaotic beauty. On one side, a smooth, continuous ribbon flows out. On the other side, a stitching machine is furiously looping back and forth, creating fragments. Eventually, the RNA primers, the temporary handles, must be removed. Another enzyme scrubs them away and replaces them with real DNA. Finally, an enzyme called ligase comes in. Ligase is the glue. It finds the tiny gaps between the Okazaki fragments and seals them shut. It smooths out the backbone. The result? 2 identical double helices. This method is called semi-conservative replication. It is semi-conservative because each new DNA molecule consists of one old strand from the original parent and one new strand that was just built. You never destroy the past. You build the future upon it. Every DNA molecule in your body holds half of a molecule that existed before you were born. But what about mistakes? With billions of letters to copy, surely the polymerase makes errors. It does. It might accidentally put a T where a C should be, but nature is wise. DNA polymerase has a delete key. It proofreads its own work. If it adds the wrong base, it senses the shape is wrong, the puzzle piece doesn't fit, and it backs up, cuts out the wrong letter, and tries again. Thanks to this proofreading, the error rate is incredibly low, about 1 mistake in every 10 billion nucleotides. However, some mistakes do slip through. These are mutations. We often think of mutations as bad as the cause of cancer or genetic disease, and often they are. But they are also the engine of evolution. Without these tiny copying errors, life would never change. There would be no variation. There would be no adaptation. We would all still be single-celled organisms floating in a primordial sea. The imperfection of the copy is the source of our diversity. Consider also the problem of the ends. Linear chromosomes have a flaw. Because the polymerase needs a primer to start, it can never quite copy the very tip of the lagging strand. Every time a cell divides, the DNA gets a tiny bit shorter. To protect the important genes, the ends of your chromosomes are capped with long stretches of nonsense DNA called telomeres. They are like the plastic tips on shoelaces, aglets, that keep them from fraying. As you age, your telomeres get shorter and shorter. Eventually, they are gone, and the cell can no longer divide safely. It enters a state of retirement called senescence. This is one of the fundamental clocks of aging. The limit of your life is written in the shortening of your strands. But tonight, do not worry about the limit. Focus on the abundance. Right now, in the quiet dark of your body, this miracle is performing millions of times over. The zipper opens, the engine builds, the glue seals, 1 becomes 2. It is a process of infinite hope. It is the promise that life will continue. It is the mechanism of renewal. You are being renewed as you sleep. Your skin is replacing itself. Your gut is healing itself. Your immune system is restocking its armies. All of it relies on the quiet, rhythmic clicking of the polymerase. Visualize the two new helices coiling up. They are perfect twins. They separate, ready to inhabit two new cells. The library has been doubled. The knowledge is safe. Now that the master copy has been duplicated, the cell can think about using it. The DNA is the archive, but it is stuck in the nucleus. To build a body, the information must get out. It needs a messenger. Our next lesson will explain how the cell reads the books. We will watch the code be transcribed into a mobile format that can travel to the factories. Lesson 11. Transcription and RNA Processing. In our last lesson, we stood inside the nucleus and watched the DNA replicate itself. We saw the library double its collection so that life could continue in a new cell. But simply possessing a library is not enough. A book that sits on a shelf, unread, is just paper and ink. For the information to have meaning, it must be used. It must be read. But there is a problem. The DNA is too precious, too large, and too fragile to leave the safety of the nuclear vault. It is the master copy. If you were an architect with the only existing blueprints for a cathedral, you would not bring those original blueprints to the dusty, muddy construction site. You would leave them in the safe. Instead, you would make a copy, a temporary, disposable sketch, and send that to the workers. In the cell, this process of copying a specific instruction is called transcription. The disposable sketch is a molecule called RNA. Let us look at this new character, ribonucleic acid. It is very similar to DNA, but with three crucial differences. First, its sugar is slightly different. It is ribose instead of deoxyribose. It has one extra oxygen atom. Second, it is usually single-stranded. It does not form a double helix. It is a single, flexible ribbon. Third, and most importantly, it changes the alphabet. It keeps adenine, cytosine, and guanine, but it replaces thymine T with a new base called uracil U. Wherever DNA would have a T, RNA has a U. It is a subtle shift in dialect, like changing a letter in a word to mark it as a copy. So, how does the cell write this message? The process begins when the cell receives a signal that it needs a specific protein. Perhaps the body needs insulin. The cell must find the specific book in the DNA library, the insulin gene. It does not unzip the entire genome. That would be a waste. It opens only the specific section it needs. The hero of this story is an enzyme called RNA polymerase. Unlike the DNA polymerase we met last time, which was a heavy-duty construction machine, RNA polymerase is like a focused scribe. It binds to the DNA at a specific start here sign called a promoter. This promoter is a sequence of DNA that tells the enzyme the gene begins right after this point. Once attached, the RNA polymerase pries the two DNA strands apart, creating a small bubble. It moves down the template strand, reading the code. If it sees a G, it adds a C to the new RNA strand. If it sees an A, it adds a uracil U. It strings these nucleotides together, creating a long growing tail of RNA that trails behind it. This is the molecule known as messenger RNA, or mRNA. It is the message that will be carried to the protein factories. Getty images. Visualize this enzyme sliding down the DNA. The double helix unzips in front of it and zips back up immediately behind it. The DNA is not permanently changed. It is just being read. The RNA strand peels off, dangling free. Eventually, the enzyme hits a stop sequence in the DNA, the terminator. The scribe lifts its pen, the RNA polymerase detaches, and the new strand of mRNA is released. But the story is not over. What comes off the DNA is not yet ready for the world. We call it pre-mRNA. It is a rough draft. It is raw footage. Before it can leave the nucleus, it must be edited and armored. This is RNA processing. First, we must protect the ends. The cytoplasm outside the nucleus is a dangerous place. It is filled with enzymes called exon nucleases that love to chew up loose RNA. To prevent the message from being destroyed before it is read, The cell adds a helmet and a tail. On the front end, the 5' end, a special cap is added. This is a modified guanine nucleotide. It acts like a hard hat. It protects the start of the message and also helps the ribosome attach later on. On the back end, the 3' end, the cell adds a long string of adenine nucleotides, hundreds of them. This is called the poly-A tail. It acts like a fuse. As the mRNA floats in the cytoplasm, the enzymes nibble away at the tail. The longer the tail, the longer the message survives. It is a built-in timer for the lifespan of the instruction. Second, we must edit the content. Here is one of the strangest facts of eukaryotic biology. Most of your genes are full of nonsense. Imagine reading a book where every meaningful sentence is interrupted by 10 pages of gibberish. That is your DNA. The meaningful parts of the gene, the parts that actually code for protein, are called exons because they are expressed. The nonsense interruptions are called introns because they are in between. Before the mRNA leaves the nucleus, the introns must be cut out. This process is called splicing. Shutterstock explore. A massive complex called the spliceosome, made of proteins and small RNA molecules, assembles on the strand. It grabs the ends of the intron, loops it out, cuts it, and stitches the 2 exons together. It is a precise cinematic edit. If the spliceosome misses by even a single letter, the entire message becomes garbage. Why does the cell have introns at all? It seems wasteful. The answer lies in versatility. By cutting the same gene in different ways, keeping some exons and throwing away others, the cell can make different proteins from the same single gene. This is called alternative splicing. It explains how humans can be so complex with so few genes. We have about 20,000 genes, the same number as a microscopic worm. But thanks to alternative splicing, we can rearrange those 20,000 genes to make over 100,000 different proteins. It is not the number of books in the library that matters. It is how you edit them. Once the cap is on, the tail is added, and the introns are spliced out, the molecule is now mature mRNA. It is ready. It approaches the nuclear envelope. It finds a nuclear pore, one of the guarded gates we saw earlier. It presents its credentials, the 5 cap. The gate opens, and the messenger slips out into the cytoplasm. This moment is critical. It is the transition from the potential to the actual. Inside the nucleus, the gene was just a possibility. Outside, in the cytoplasm, it becomes a command. Think of the real-world power of this molecule. The mRNA vaccines that change the world work on this exact principle. They do not change your DNA. They simply introduce a piece of mRNA, a specific instruction note, into your cytoplasm. Your cells read the note, build the protein spike of the virus, and train your immune system. They utilize the natural transcription and translation pathways that have existed for billions of years. As you drift in the darkness, visualize these ribbons of code flowing out of your nucleus. They are like streamers of light. They carry the orders that keep you alive. Build more skin, digesting dinner, dreaming. Every function of your body begins with this transcription. The silent scribe writing the notes that become your reality. The message is now out in the wild. It is floating in the cytosol, but it is still just code. It is a language of nucleotides. The cell needs a language of amino acids. It needs to translate the blueprint into a building. To do this, it needs a translator. It needs a factory. Our next lesson will take us to the ribosome. We will watch the final act of the central dogma, where the code becomes flesh. Lesson 12. Translation and Protein Synthesis. We have followed the path of information from the vault of the nucleus to the open sea of the cytoplasm. We have seen the master code of DNA transcribed into the working message of RNA. But a message is not a structure. A blueprint is not a building. You cannot live on instructions alone. You need substance. You need action. This brings us to the most complex and vital step in the central dogma of biology: translation. Why do we call it translation? Think about language. DNA and RNA are written in the language of nucleotides, the 4 letters we know so well. But the machines of the cell, the proteins, are built in a completely different language. They are written in the language of amino acids. There are 20 distinct amino acids. each with different chemical properties. To turn the code into a creature, the cell must translate from the 4-letter alphabet of nucleic acids to the 20-letter alphabet of proteins. This translation takes place at the ribosome. Visualize this organelle. It is not a membrane-bound sac like the others we have seen. It is a solid molecular machine made of ribosomal RNA and proteins tangled together. It consists of 2 parts, a small subunit and a large subunit. Imagine a hamburger bun. The small subunit is the bottom bun. The large subunit is the top bun. Most of the time, they float separately in the cytoplasm. But when an mRNA message arrives, they come together, clamping down on the RNA strand like a vice. The ribosome is the workbench. The mRNA is the instruction tape running through it. But where are the materials? Who brings the bricks? This is the job of transfer RNA, or tRNA. Visualize the tRNA molecule. It is a single strand of RNA folded up into a shape that looks like a 3-leaf clover. It is the bridge between the 2 languages. At the top of the clover, it carries a specific amino acid, perhaps a molecule of methionine, or proline, or serine. At the bottom of the clover, it has a set of 3 exposed nucleotides called the anticodon. The system works on a rule of 3s. The ribosome reads the mRNA message 3 letters at a time. This triplet is called a codon. If the mRNA says AUG, that is a codon. The tRNA that matches it must have the complementary anticodon UAC. And that specific tRNA always carries the amino acid methionine. It is a perfect matching game. The codon calls for a specific shape. The tRNA fits that shape and delivers exactly the right cargo. Let us watch the process unfold in 3 slow, deliberate stages. First is initiation. The small ribosomal subunit finds the 5 cap of the mRNA, the protective helmet we added last lesson. It slides down the ribbon until it finds the specific start sequence, AUG. This is the universal start signal. A tRNA carrying methionine binds to it. Then, the large subunit clamps down on top. The machine is assembled. The green light is lit. Second is elongation. This is the cycle of creation. The ribosome has 3 slots inside it, which we call the A site, the P site, and the E site. You can remember them as arrival, parking, and exit. The ribosome sits on the start codon in the P site. A new tRNA arrives at the A site, matching the next codon on the tape. It slots in perfectly. Now, a reaction occurs. The ribosome takes the amino acid from the first tRNA and bonds it to the amino acid on the new tRNA. This bond is called a peptide bond. It is the strongest link in biology. The first tRNA is now empty. The second tRNA is holding a chain of 2 amino acids. The ribosome then shifts forward. It moves exactly 3 spaces, 1 codon down the mRNA tape. This movement is called translocation. It is a ratcheting motion. Click, shift. The empty tRNA is moved to the E site, exit, where it is ejected to go find another amino acid. The tRNA holding the chain moves to the P site. The A site is now open, ready for the next delivery. Visualize this cycle repeating. Arrival, bond, shift, exit. Arrival, bond, shift, exit. It happens with hypnotic regularity. The polypeptide chain grows longer and longer, emerging from the top of the ribosome like a ribbon growing out of a chimney. The ribosome reads the tape, GGC. It calls for glycine. The tRNA brings glycine. Click. The ribosome reads UCA. It calls for serine. The tRNA brings serine. Click. Amino acid by amino acid. The chemical language is translated into physical reality. Finally, we reach the 3rd stage, termination. The ribosome encounters a stop codon UAA, UAG, or UGA. There are no tRNAs that match these stops. Instead, a protein called a release factor binds to the A site. It acts like a pair of molecular scissors. It cuts the bond between the finished protein chain and the last tRNA. The ribosome falls apart. The 2 buns separate. The mRNA floats away, perhaps to be read again, or perhaps to be recycled. The new protein is born. But the story is not quite finished. What comes out of the ribosome is just a linear string, a polypeptide. It is like a piece of yarn. To become functional, it must fold. As the chain emerges into the water of the cytoplasm, the laws of chemistry take over. Hydrophobic amino acids want to hide from the water, so they curl into the center. Positive charges stick to negative charges. Sulfur atoms bind to other sulfur atoms, creating strong bridges. Spontaneously, or sometimes with the help of chaperone proteins, the string twists, bends, and spirals into a complex, three-dimensional shape. It might become a globular enzyme, ready to digest sugar. It might become a long, fibrous strand of collagen, ready to support skin. It might become a pore in the membrane, ready to let ions flow. This is the tertiary structure. The shape is the function. If the translation had even one error, one wrong amino acid, the folding might fail, and the protein would be useless or even dangerous. Consider the universality of this code. The table of codons which triplet equals which amino acid is nearly identical in every living thing on Earth. If you took the gene for a glow-in-the-dark protein from a jellyfish and put it into a bacteria, the bacteria's ribosomes would read it perfectly and build the protein. The bacteria would start to glow. The hardware is compatible because we are all built by the same manufacturer, evolution. This process is also the battlefield of medicine. Many antibiotics, like erythromycin or tetracycline, work by targeting ribosomes, but they are smart weapons. They are shaped to jam the gears of bacterial ribosomes, which are slightly different from human ribosomes. When you take these drugs, they stop the bacteria from building proteins. Without new proteins, the bacteria cannot grow, and the infection dies. Your own ribosomes, different in shape, keep working peacefully. As you lie here, your body is engaged in this act of massive construction. Billions of ribosomes are clicking and shifting. They are building the antibodies that protect you. They are building the enzymes that will digest your breakfast tomorrow. They are building the neurotransmitters that will form your dreams tonight. You are being constantly remade. You are not a static object. You are a river of synthesis. Visualize the chain growing, link by link, strong, perfect. The code has become matter. The thought has become a thing. There is a profound satisfaction in this completion. The task is done. The protein is released to do its work. But as the proteins build the cell and the cell grows larger, it eventually faces a limit. A cell cannot grow forever. It becomes too large to feed itself. To survive, it must do something drastic. It must replicate not just its DNA, but its entire self. It must divide. Our next lesson will take us from the molecular to the cellular. We will watch the grand spectacle of division. Lesson 13. The cell cycle and mitosis. We have arrived at the climax of the cellular life. This is the cell cycle and the dramatic process of mitosis. Most of a cell's life is not spent dividing. It is spent in a state of quiet industry called interphase. If you were watching a cell under a microscope, interphase might look boring. The cell is just sitting there. But this is an illusion. Beneath the surface, the cell is furiously preparing. Interphase is divided into 3 chapters. First is the G1 phase, or gap 1. This is the time of growth. The cell eats, breathes, and builds new organelles. It stocks up on materials. It decides whether the time is right to divide. Next comes the S phase, or synthesis. This is the miracle we watched in Lesson 10. The DNA unzips and replicates. The single library becomes 2 identical libraries. Finally, there is the G2 phase, or gap 2. This is the final check. The cell proofs the DNA for errors. It builds the specialized proteins needed for the physical act of splitting. It is the deep breath before the plunge. Now, the preparation is over. The curtain rises on the M phase. Mitosis. Mitosis is the division of the nucleus. It is a strictly choreographed ballet in 4 acts. Visualize the cell now, large and ready. The nucleus is dark and heavy in the center. Act 1. Prophase. The first sign of change is in the DNA itself. Normally, DNA is a loose, tangled mess called chromatin, like a bowl of spaghetti. But you cannot divide a bowl of spaghetti evenly without knotting and breaking the noodles. So, the cell condenses the DNA. The long strands coil up tighter and tighter, winding around histones, packing down into thick, distinct shapes. These are the chromosomes. They look like tiny X's. Each X is actually two identical copies of DNA called sister chromatids, stuck together at the center by a button called a centromere. As the chromosomes condense, the nuclear envelope, the protective vault we have admired for so long, begins to disintegrate. The walls dissolve. The library is open. Simultaneously, the cell's skeleton rearranges itself. The centrosomes, the organizers of the microtubules, move to opposite ends of the cell, like the north and south poles of a planet. Between them, they spin a web of fibers called the mitotic spindle. Shutterstock Explore Act 2. Metaphase. This is the moment of alignment. The spindle fibers reach out across the cell and grab onto the centromeres of the floating chromosomes. They begin a tug of war. One fiber pulls left, another pulls right. The chromosomes are jostled and pushed until they line up perfectly in the center of the cell. We call this the metaphase plate. It is not a physical structure, but a plane of alignment, like soldiers standing at attention. This is a critical checkpoint. The cell pauses. It waits. It checks to make sure every single chromosome is attached to a fiber. If even one is loose, the process halts. The cell will not proceed until perfection is achieved. It is a moment of breathtaking symmetry. 46 X-shapes, lined up in a row, ready to part ways. Act 3. Anaphase. The signal is given. The glue holding the sister chromatids together dissolves. Snap. In an instant, the X-shapes are pulled apart. The sisters become strangers. The spindle fibers shorten, reeling in the DNA like a fisherman reeling in a catch. The chromosomes are dragged rapidly toward the opposite poles. They look like V-shapes now, their arms trailing behind them as they are pulled through the thick cytoplasm. The cell elongates, stretching out into an oval. This is the physical separation of the library. One complete set of books goes to the north. One complete set goes to the south. Nothing is left behind. Act 4. Telophase. The journey ends. The chromosomes arrive at the poles. The tension releases. Now, the cell must undo the drama of prophase. The tightly coiled chromosomes begin to relax, unspooling back into the loose, comfortable spaghetti of chromatin. New nuclear envelopes begin to form around the two clusters of DNA. The vault is rebuilt. The spindle fibers, their job done, dissolve back into the cytoplasm. We now have one cell with two nuclei, two brains in one body. But the process is not complete. The nucleus has divided, but the body has not. This final step is called cytokinesis. In animal cells like yours, this happens through a process called cleavage. Visualize a ring of actin filaments forming around the center of the cell, right where the chromosomes used to be lined up. This ring is like a drawstring on a pouch. It begins to tighten. It pulls the membrane inward, creating a groove called the cleavage furrow. It squeezes tighter and tighter. The cell is pinched in the middle. The waste becomes thinner and thinner until pop. The connection is severed. Where there was one large cell, there are now two smaller, identical daughter cells. In plant cells, the process is slightly different because of the rigid cell wall. They cannot pinch. Instead, they build a new wall from the inside out. Vesicles filled with cellulose line up in the center and fuse together, creating a cell plate that grows until it divides the room in two. Reflect on the significance of this event. Every cell in your body is the result of this dance. You began as a single zygote, one cell. That cell performed mitosis to become 2, then 4, then 8, then billions. Mitosis is how you grew from an infant to an adult. Mitosis is how you heal. When you cut your skin, the cells on the edge of the wound wake up. They receive signals to leave the quiet G1 phase and enter the cycle. They divide and crawl across the gap, knitting your skin back together. Mitosis is the engine of renewal. Your red blood cells live for only about 4 months. Your stomach lining replaces itself every few days. You are constantly dying and being reborn, cell by cell. But this power must be controlled. The cell cycle is not a runaway train. It is governed by strict checkpoints. At the end of G1, the cell asks, "Am I big enough? Is the environment safe?" At the end of G2, the cell asks, "Is the DNA copied correctly?" At metaphase, the cell asks, "Are the chromosomes lined up?" These checkpoints are guarded by special proteins called cyclins and CDKs. They are the traffic lights of the cell. When these traffic lights fail, the consequences are dire. If a cell ignores the red light, if it divides when it is too small, or when its DNA is damaged, it becomes a danger to the community. This is the origin of cancer. Cancer is essentially mitosis gone wrong. It is a disease of uncontrolled division. The cancer cell is a cell that has forgotten how to stop. It rushes through the checkpoints, dividing endlessly, piling up into a tumor, hoarding resources. It is a rebellion against the order of the body. But tonight, in your healthy body, the order is maintained. The traffic lights are working. The sentries are at their posts. The dance is precise. Visualize the clean separation, one becoming two, the feeling of duplication, the feeling of abundance. There is a sense of freshness in the daughter cells. They are new. They are ready to begin their own lives, to grow, to work, and eventually to divide again. It is an infinite chain of life, stretching back to the beginning of time and forward into the future. As you drift into sleep, imagine this rhythm of renewal washing over you. Divide and grow, divide and heal, divide and become. But there is a dark side to division. Not all death is accidental. Sometimes, for the good of the organism, a cell must choose to die. It must make the ultimate sacrifice. Our next lesson will explore the balance between life and death. We will learn the difference between a messy end and a noble exit. Lesson 14. Apoptosis and Necrosis. Tonight, we explore the necessary end. We will learn that death, in the cellular world, is not always a tragedy. Often, it is a deliberate, noble choice made for the greater good. In this lesson, we will distinguish between the two faces of cellular death, the orderly exit known as apoptosis and the chaotic accident known as necrosis. Let us begin with the concept of the good death. The word apoptosis comes from the ancient Greek. It describes the falling of leaves from a tree in autumn or the dropping of petals from a flower. It is a poetic name for a gentle process. It implies a natural, seasonal shedding. Apoptosis is programmed cell death. It is a suicide sequence encoded in the cell's own DNA. Every cell in your body carries the instructions for its own demise, locked away in a safety box, waiting for the command to open it. Why would a cell choose to die? There are many reasons. Perhaps the cell is old and its machinery is worn out. Perhaps it is infected by a virus and wants to stop the spread. Perhaps its DNA has been damaged beyond repair, and it fears becoming cancerous. In these cases, the cell acts with supreme altruism. It realizes it is a danger to the collective, and so it initiates the protocol. Shutterstock Explore. Let us visualize this process. It is a controlled demolition. Imagine a building being taken down in the center of a busy city. You cannot just blow it up. The debris would hurt the neighbors. You must take it down floor by floor, carefully wrapping the windows, containing the dust. When the apoptotic signal is triggered, a family of executioner enzymes called caspases wakes up inside the cell. These caspases act like precise scissors. They begin to cut the proteins of the cytoskeleton, the actin and tubulin we learned about earlier. The cell loses its shape. It rounds up and shrinks, pulling away from its neighbors. It cuts its connections, the desmosomes and junctions dissolve. It is saying goodbye. Next, the caspases enter the nucleus. They chop up the DNA into small, uniform fragments. The library is shredded to prevent it from ever being used again. As the cell collapses inward, the membrane begins to bulge and pinch off. This is called blebbing. The cell breaks itself into small, neat packages called apoptotic bodies. Crucially, the membrane never bursts. The dangerous contents, the digestive enzymes of the lysosome, the acidic fluids, are kept safely sealed inside these little bags. Finally, a signal is sent out to the immune system. Come and eat me. A macrophage, a large white blood cell, arrives and gently engulfs the apoptotic bodies. The cell vanishes without a trace. There is no inflammation, there is no pain, there is only a quiet disappearance, leaving room for a new, healthy cell to take its place. Shutterstock. Explore now. We must contrast this with the bad death. This is necrosis. If apoptosis is a controlled demolition, necrosis is a bomb going off. It is accidental, traumatic, and messy. Necrosis is not a choice. It is caused by external injury, a burn, a crush, a poison, or a lack of oxygen, ischemia. Imagine a heart attack. A blood vessel is blocked. The heart muscle cells are starved of oxygen. The mitochondria can no longer make ATP. Without energy, the ion pumps in the membrane stop working. Sodium rushes in, followed by water. The cell does not shrink, it swells. This is called oncosis. It bloats like an overfilled water balloon. Eventually, the membrane cannot hold the pressure. It ruptures. All the toxic contents of the cell, the acids, the enzymes, the raw chemicals spill out into the surrounding tissue. This is a disaster for the neighbors. The release of these chemicals triggers a massive alarm. The immune system rushes in, not to clean up quietly, but to fight. This causes inflammation. The area becomes red, hot, swollen, and painful. The death of one cell can kill the cells around it, creating a domino effect of destruction. Let this distinction settle in your mind. Apoptosis, shrinkage; order, encapsulation, silence. Necrosis, swelling, chaos, rupture, alarm. One sustains life, the other threatens it. Consider the role of the good death in your own history. You are alive today because of death. When you were an embryo in the womb, your hand started as a solid paddle. You did not have fingers, you had a web of tissue. To create your hand, the cells in the webbing between your future fingers had to die. They triggered apoptosis in a wave, receding like the tide to reveal the separate digits. If you have fingers to wiggle right now, thank the cells that sacrificed themselves to carve them out. The same is true for your brain. As an infant, your brain produced far too many neurons. As you learned and experienced the world, the connections you used were strengthened, and the neurons you didn't use were pruned away by apoptosis. You carved your intelligence out of the excess. You are a sculpture, created by the chipping away of the unnecessary. Real-world medicine is deeply concerned with this balance. Many diseases are essentially a problem of the math of death. In neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's, the problem is too much apoptosis. Cells in the brain are triggering the suicide protocol when they shouldn't. They are dying prematurely, stealing memories and function. On the other hand, in autoimmune diseases, the problem is often too little apoptosis. Immune cells that react against your own body should be killed off in the thymus gland. If they fail to die, if they refuse the order, they escape into the blood and attack your joints or your nerves. And then, there is the greatest failure of all. What happens when a cell that is dangerous, a cell with mutated DNA, refuses to die? Normally, the protein p53, known as the guardian of the genome, scans the DNA. If it finds damage, it pauses the cell cycle to fix it. If it can't fix it, p53 orders apoptosis. It executes the cell to save the body. But if p53 is broken, if the safety mechanism fails, the damaged cell lives. It divides. It creates a daughter that is also damaged. They pile up. They ignore the signals to stop. They ignore the signals to die. This is the seed of a tumor. We see now that life is a tightrope walk. You need enough death to stay healthy, to shape your body, and to remove the dangers, but not so much death that you lose your function. Your body is walking this tightrope right now. In your sleep, it is balancing the equation perfectly. Old skin cells are sloughing off apoptosis. New skin cells are rising up mitosis. The balance is maintained. Visualize the autumn leaf again. It does not fall because the tree is broken. It falls because the tree is wise. By shedding the leaf, the tree conserves water for the winter. It protects the whole. See the cells of your body acting with this same wisdom. They are not afraid of the end. They accept it as part of their duty. They serve the greater organism, they serve you. There is a deep peace in this acceptance. The cycle is complete. The beginning implies the end, and the end allows for a new beginning. You can let go of the day, just as the cell lets go of its life. You can let go of your thoughts. You can let go of your tension. Let it drift away like a falling leaf. Down, down, into the dark, rich soil of sleep. But we mentioned a danger. We mentioned a cell that refuses to die. A cell that breaks the contract. Our next lesson will explore this rebellion. We will look at what happens when the checks and balances fail, and the cycle runs wild. Lesson 15. Cancer Biology. We have spent 14 lessons exploring the exquisite order of life. We have seen how the DNA is protected, how the proteins are built with precision, and how the cell cycle is regulated by strict checkpoints. We have seen that the body is a society of trillions of cells, all working together for the common good. But in any vast society, there is the potential for rebellion. Tonight, we must face the reality of what happens when the rules are broken. We turn our gaze to the phenomenon known as cancer. It is a word that carries great weight in the waking world, often evoking fear. But here, in the safety of this learning space, we will look at it simply as biology. We will deconstruct it. We will understand it. Cancer is not an invader from the outside, like a virus or a bacteria. It is not other. It is us. It is a cell of your own body that has forgotten its duty. It is a cell that has chosen the self over the whole. At its most fundamental level, cancer is a disease of the cell cycle. It is uncontrolled mitosis. Recall the traffic lights we discussed, the checkpoints that tell a cell when to stop dividing. A cancer cell runs the red lights. It ignores the stop signs. It divides and divides and divides, regardless of whether the body needs new cells or not. How does this happen? The origin lies in the library, in the DNA. Cancer begins with mutation, a change in the code. A single mutation is usually not enough to cause cancer. Your body has backup systems. But over time, if a cell accumulates multiple specific mutations, the safeguards fail. To understand these mutations, biologists use the analogy of a car. For a car to drive safely, it needs two things: a working gas pedal and working brakes. The cell is the same. First, let us look at the gas pedal. In a normal cell, there are genes called proto-oncogenes. These are good genes. They code for proteins that tell the cell to grow and divide when necessary, for example, to heal a wound. They are the gas pedal. But if a mutation occurs in one of these genes, it can become stuck in the on position. It becomes an oncogene. Imagine a gas pedal pressed all the way to the floor and welded there. The engine revs. The cell receives a constant screaming signal: divide, divide, divide. It does not matter if there is no space. The signal is relentless. Shutterstock. Second, let us look at the brakes. These are the tumor suppressor genes. Their job is to slow down cell division, to repair DNA mistakes, or to tell the cell to die, apoptosis, if the damage is too great. The most famous of these is a protein called p53, often called the guardian of the genome. p53 is the master inspector. It scans the DNA. If it finds an error, it halts the factory. It prevents the car from moving until the repair is made. But if a mutation breaks the p53 gene, the brakes are cut. The inspector is blindfolded. Now, consider the terrifying math. You have a cell with a stuck gas pedal, an oncogene, and cut brakes, a mutated tumor suppressor. The car is speeding down the highway with no way to stop. This is the birth of a tumor. As this rogue cell divides, it creates a clone army. A cluster of cells forms. If these cells remain clustered together and do not invade other tissues, we call this a benign tumor. It is a lump, but it is contained. It can usually be removed. But if the cells accumulate further mutations, they become malignant. This is true cancer. Malignant cells develop terrifying superpowers that normal cells do not have. Normal cells exhibit contact inhibition. When they bump into a neighbor, they stop growing. They respect personal space. Cancer cells have lost this respect. They climb over one another, piling up into chaotic mounds. Normal cells exhibit anchorage dependence. They must be attached to the extracellular matrix, the soil, to survive. If they float free, they self-destruct. Cancer cells have learned to survive without an anchor. They can float. But a tumor cannot grow very large on its own. It needs food. It needs oxygen. This leads to one of the most sinister tricks of cancer, angiogenesis. The tumor sends out chemical signals, SOS messages, that mimic the signals of a starving tissue. These signals drift to the nearby blood vessels and trick them. The body, thinking it is helping a wound, grows new blood vessels directly into the tumor. The tumor has hijacked the body's supply lines. It feeds on the glucose and oxygen meant for healthy organs, growing larger and stronger. Shutterstock explore. Then comes the final and most dangerous phase, metastasis. Because the cancer cells have lost their adhesion molecules, their desmosomes and tight junctions, they are loose. They can detach from the main tumor. They crawl through the tissue. They squeeze through the walls of the new blood vessels and drop into the bloodstream. Visualize a single cancer cell floating down the dark river of a vein. It is a traveler. It is carried by the current to a distant part of the body, the lungs, the liver, the brain. It exits the bloodstream and takes root in this new soil. It begins to divide again. This is why cancer is so difficult to treat. It is not just one target, it becomes many targets, hidden in different landscapes. However, understanding this biology is the key to fighting it. Every treatment we have is based on the lessons we have learned tonight. Because cancer cells divide rapidly, we use chemotherapy drugs that target dividing cells. They might attack the mitotic spindle or the DNA replication enzymes. The side effects occur because other fast-dividing cells, like hair follicles and gut lining, are caught in the crossfire. Because cancer cells need blood, we use angiogenesis inhibitors to starve the tumor. Because cancer cells hide from the immune system, we use immunotherapy to rip off their disguises and teach the white blood cells to attack them. We are entering a new era. We are moving away from slashing and burning and toward precision medicine. We can now sequence the DNA of a patient's tumor, find the specific break that is broken or the specific pedal that is stuck, and design a drug to fix just that one part. There is a philosophical lesson here as well. The cancer cell is immortal. It has reactivated an enzyme called telomerase, which rebuilds the tips of its chromosomes. It can divide forever. It refuses to age. It refuses to die. But in doing so, it destroys the host. By seeking immortality for itself, it ensures the death of the whole. It is a reminder that biological limits, aging, checking, stopping, dying, are not punishments. They are the safeguards of life. We die so that the organism may live. We accept limits so that the community can thrive. As you lie here, your body is patrolling. Your immune system is watching. Your p53 proteins are scanning the library, ensuring the books are in order. You are protected by billions of years of evolutionary wisdom. The system is vigilant so that you can be at peace. Visualize the order returning. See the rogue cells being recognized and removed. See the traffic lights turning red and the cells obediently stopping. See the harmony of the society restored. The rebellion is quelled. The peace of the body is maintained. We have now explored the life cycle of the individual cell, from birth to death, and even into disease. But life does not end with the individual. Life is a chain. For the species to survive, the information must be passed to a new generation. We must move from the somatic, the body, to the genetic, the future. Our next lesson will look at the special type of division that allows for reproduction. We will learn how the deck of genes is shuffled to create a unique new being. Lesson 16. Meiosis and Sexual Reproduction. In our previous lessons, we watched the cell clone itself. We saw mitosis, where one cell makes an exact photocopy of its library. This is perfect for growth. It is perfect for healing a wound. But it is not how we make a new human being. If we simply took a cell from your father and fused it with a cell from your mother, the math would be disastrous. Your father has 46 chromosomes. Your mother has 46 chromosomes. If they combined, you would have 92. The next generation would have 184. The cells would burst with DNA. The chaos would be instant. To solve this problem, life invented a special type of division, a mathematical magic trick that cuts the number of chromosomes exactly in half. This is meiosis. It is the gateway to sexual reproduction. To understand this, we must first understand the concept of the homologous pair. Look at the nucleus of any cell in your body, except the sperm or egg. You have 46 chromosomes. But they are not 46 random, unique books. They are 23 pairs of books. For every chromosome type, let's say chromosome number 1, you have 2 copies. One copy came from your mother. The other came from your father. They cover the same topics. They both have genes for eye color, for height, for blood type, but they might tell different stories. Your dad's version might say blue eyes, while your mum's version says brown eyes. We call these match sets homologous chromosomes. A cell with 2 sets is called diploid, or 2N. A cell with only 1 set is called haploid, or N. The goal of meiosis is to turn a diploid cell with 46 chromosomes into a haploid cell with 23. These special haploid cells are the gametes, the sperm and the egg. Let us visualize this grand reduction. It happens in the reproductive organs, the testes and the ovaries. It begins, like mitosis, with interphase. The DNA replicates. We start with 46 chromosomes, each consisting of 2 sister chromatids. But then, the cell enters a process so complex it requires 2 full rounds of division, meiosis I and meiosis II. Meiosis I is where the magic happens. The curtain rises on prophase I. The chromosomes condense and become visible. But then, they do something they never do in mitosis. They find their partners. The chromosome 1 from your dad floats through the nucleus and finds the chromosome 1 from your mom. They align perfectly, length for length, gene for gene. This intimate pairing is called synapsis. While they are hugged tight together, they perform a critical act called crossing over. Shut up, visualize 2 ribbons, one blue and one red, twisted around each other. The enzymes snip the ribbons and swap the ends. A piece of the blue ribbon is attached to the red ribbon, and vice versa. They are trading genes. This is profound. Before this moment, that chromosome was purely your father's. Now, it is a hybrid. It is a unique mixture that has never existed before in the history of the universe. This is the source of genetic variation. You are shuffling the deck of your ancestors. The cell then moves to metaphase I. The homologous pairs, still hugging, line up in the center of the cell. But how they line up is random. The dad chromosome might be on the left, or it might be on the right. This is called independent assortment. With 23 pairs, there are over 8 million possible arrangements. Then comes anaphase I. The pairs are pulled apart. This is the moment of reduction. The dad pair goes to one pole. The mom pair goes to the other. But notice, the sister chromatids, the copies, are still stuck together. We are separating the partners, not the copies. The cell divides. We now have 2 cells. Crucially, these cells are now haploid. They only have 23 chromosomes, but each chromosome is still double-stranded. Now, without stopping to copy the DNA again, the cells dive straight into meiosis II. This looks exactly like mitosis. The chromosomes line up in the center. The spindle fibers grab on. Anaphase II occurs. The sister chromatids. The copies are finally ripped apart. The cells divide again. Let us count the result. We started with 1 diploid cell. We ended with 4 haploid cells. In a male, these 4 cells will all become sperm. In a female, typically one becomes the egg, and the other 3 are discarded as polar bodies, sacrificing their cytoplasm to ensure the egg has enough fuel to survive. Why go through all this trouble? Mitosis is simple. Cloning is easy. Bacteria do it all the time. Why sex? The answer is variation. If you clone yourself, your offspring has your exact strengths, but also your exact weaknesses. If a virus comes along that can kill you, it will kill all your clones. The species would go extinct. But through meiosis, through the shuffling of crossing over and independent assortment. You create a child that is different. Maybe they have a slightly different immune system. Maybe they are slightly faster or taller. Variation is the insurance policy of life. It creates a diverse population that can survive a changing world. It is the fuel for natural selection. Think of a deck of cards. If you just photocopy the deck, mitosis, you always play the same hand. But if you shuffle two decks together, meiosis and fertilization, the number of possible hands you can deal is virtually infinite. You are one of those hands, a unique combination that has never been played before. However, this complexity comes with a risk. The dance of the chromosomes is intricate, and sometimes the dancers trip. This is called non-disjunction. Visualize anaphase. The fibers pull, but the chromosomes refuse to separate. They stick together. Both copies get dragged to the same side. This results in a gamete with an extra chromosome, or one missing a chromosome. If a sperm with 24 chromosomes fertilizes an egg with 23, the resulting child will have 47. The most common example of this is Down syndrome, also known as trisomy 21. The individual has 3 copies of the 21st chromosome instead of 2. It is a reminder that our biology is a physical mechanism, subject to mechanical error. It is not perfect, but it is persistent. Explore now. Bring your focus back to the beauty of the system. Think of the moment of fertilization. The haploid sperm meets the haploid egg. 23 meets 23. The nuclei fuse. 46 is restored. The diploid state returns. This single cell, the zygote, now contains the full library needed to build a human being. But it is a new library, a revised edition, with a mix of volumes from 2 different lineages. As you rest here, understand that your cells are not performing meiosis. Meiosis is a rare event reserved for the special cells in the gonads. Your body cells, your somatic cells, are happily performing mitosis, keeping you stable. But you carry the potential for the future within you. You carry the mechanism to pass the torch. There is a sense of continuity here that spans eons. The thread of DNA stretches back through your parents, through the first humans, through the primates, through the early mammals, all the way back to the first eukaryotes who discovered that mixing genes gave them a better chance at survival. You are the current ripple in that unbroken wave. The code has been shuffled, the new individual has been defined. But how does this new individual take shape? Every cell in this new body will have the same DNA. The eye cell and the toe cell have the exact same books. So why are they so different? Why does one build a lens and the other build a nail? The secret lies not in having the books, but in choosing which ones to read. Our next lesson will explore the control panel of the genome. We will learn how the cell turns genes on and off to create the complexity of a whole organism. Lesson 1. Lesson 17. Regulation of gene expression. We have reached a pivotal moment in our understanding. We have learned that every single cell in your body, from the clear cornea of your eye to the tough enamel of your tooth, contains the exact same library. The DNA in your liver is identical to the DNA in your brain. They hold the same 20,000 instructions. They have the same potential, and yet a liver cell is nothing like a brain cell. One is a chemical factory, the other is an electrical transmitter. How can 2 entities with the exact same instructions become such radically different things? The answer lies in gene regulation. It is not about which books you have. It is about which books you choose to read. Imagine a grand piano. It has 88 keys. Every pianist has the same 88 keys. But by choosing which keys to press, how hard to press them, and when to release them, one can play a jazz standard while another plays a classical concerto. Your cells are the pianists. The DNA is the keyboard. The music they create is the phenomenon of life. In this lesson, we will explore how the cell controls this music. We will learn how genes are turned on and off. Let us begin with the simplest form of control, found in our distant cousins, the bacteria. Bacteria need to be efficient. They cannot waste energy making tools they don't need. Imagine a bacterium swimming in a bowl of milk. It needs to digest the milk sugar, lactose. To do this, it needs 3 specific enzymes. But if there is no milk around, making those enzymes is a waste. So the bacterium groups the genes for those enzymes together into a single unit, called an operon. The most famous example is the lac operon. Visualize a train track. The 3 genes are the cars of the train. At the front of the track is a switch called the operator. When there is no lactose, a protein called a repressor sits on the switch. It acts like a roadblock. The RNA polymerase, the scribe we met earlier, cannot get past. The genes are turned off. Silence. But the moment lactose enters the cell, it binds to the repressor and pulls it off the track. The roadblock is gone. The scribe rushes down the track, transcribing the genes, and the enzymes are made. It is a perfect automatic feedback loop. Simple, elegant, binary. But you are not a bacterium. You are a multicellular eukaryote. Your regulation is far more subtle and complex. You do not just need on and off. You need a little bit louder, softly now. Only on Tuesdays, only during childhood. To achieve this, your cells use a team of proteins called transcription factors. Visualize the start of a gene in a human cell. It is not just a simple switch. It is a control panel with dozens of buttons and sliders. For the RNA polymerase to even find the gene, it needs help. General transcription factors must land on the DNA first, building a landing pad for the enzyme. But this is just the baseline. To really crank up the volume, to express the gene at high levels, you need activators. These are special proteins that bind to DNA sequences called enhancers. Here is the strange geometry of the nucleus. The enhancer might be located thousands of letters away from the gene it controls. It might be miles away in molecular terms. So, the DNA bends. It loops around, bringing the distant enhancer into contact with the gene, creating a complex knot of proteins that kickstarts the process. Conversely, there are silencers. These bind to repressors that lock the gene down, preventing it from being read even if the scribe is ready. This combinational control is how you achieve complexity. To make a red blood cell, you might need activator A, activator B, and repressor C all present at the same time. To make a skin cell, you might need activator A and activator C, but not B. It is a precise chemical code that determines identity. But there is a deeper layer of control, a layer that does not change the code itself, but changes how the code is read. This is epigenetics. Epi means above or on top of. Epigenetics is the set of instructions written on top of the DNA. Think of the DNA strand. We know it is wrapped around spools called histones. If the DNA is wrapped too tightly, the genes are hidden. The scribe cannot reach them. They are silenced. If the DNA is wrapped loosely, the genes are exposed. They can be read. The cell controls this by chemically modifying the histones. It can add an acetyl group to the histone tails. This neutralizes their charge and causes the DNA to relax. Acetylation opens the book. It turns genes on, or it can add a methyl group directly to the DNA, usually to cytosine bases. This is called methylation. Methylation is like putting a heavy padlock on the gene. It condenses the chromatin, twisting it into a tight knot. It turns genes off. This epigenetic marking is crucial for differentiation. When you were an embryo, your stem cells were totipotent. They could become anything. Their DNA was open and unmethylated. But as a cell decided to become a neuron, it began to lock down the genes it didn't need. It methylated the muscle genes. It methylated the liver genes. It packed them away in the attic. By the time it became a mature neuron, only the neuron genes were left open. The cell had remembered its identity. Even when it divides, it passes these methylation patterns to its daughters. A liver cell always creates a liver cell because the other options are chemically locked away. But here is the most profound realization about epigenetics. It is not set in stone. Your environment can change your epigenetics. Stress, diet, toxins, and exercise can influence which genes are acetylated or methylated. A study of identical twins shows this clearly. When they are born, their epigenomes are identical. But as they age, as they live different lives, eat different foods, suffer different stresses, their epigenetic markers diverge. One might turn off a tumor suppressor gene that the other keeps on. One might get cancer while the other stays healthy, despite having the exact same DNA. There is even evidence that these changes can be passed down. In what is known as transgenerational epigenetic inheritance, The ghosts of your ancestors' experiences may linger in your genes. If your grandmother suffered a famine while she was pregnant, the stress signals could have altered the methylation patterns in the developing eggs of her fetus, the eggs that would eventually become you. You might be born with a metabolism shifted to hoard calories, prepared for a famine that happened 2 generations ago. It connects us to our history in a way that is both beautiful and terrifying. We are the guardians of our genome, not just for ourselves, but for the future. As you lie here, your body is managing this control panel. In your brain, genes involved in memory consolidation are being acetylated, opened up to store the lessons of the day. In your muscles, genes for repair are being activated by the rest. In your eyes, the genes for perceiving light are being silenced for the night. It is a shifting landscape of access and denial, constantly tuning the instrument of your body to match the song of your environment. There is a great peace in understanding this adaptability. You are not a robot running a fixed program. You are a responsive, dynamic being. You change, you adjust, you learn. The DNA is the score, but you are the performance. Visualize the double helix again, but now see the tiny chemical markers dotting its surface like jewels. See the DNA bending and looping, bringing distant friends together to start a conversation. See the chromatin breathing, opening to speak, closing to rest. It is a living, breathing library. We have now covered the machinery, the code, the division, and the control. But what happens when we look at the very beginning of this process? We have talked about cells deciding to become neurons or skin cells. But where do these decisions start? We must look at the cells that have not yet decided, the cells of infinite potential. Our final lesson will take us to the source of all tissues. We will look at the blank slates of biology. Lesson 1. Lesson 18. Stem cells and differentiation. In this final further session, we explore the most magical and hopeful cells in the biological world. These are the cells that have not yet decided what they want to be. They are the blank slates. They are the artists holding a brush, staring at a white canvas. We call them stem cells. To understand them, we must wind the clock back. Back before you were an adult with bones and skin and memories. Back before you were a fetus. Back to the moment of conception. When the sperm met the egg, they fused to form a single cell called the zygote. This single cell is the ultimate stem cell. We call it totipotent. The word comes from the Latin totus, meaning entire. This cell has the power to become the entire organism. It can become your brain, your heart, your toes. It can even become the placenta that feeds you. It holds within it the potential for everything that is you. As the zygote divides, 1 becomes 2, 2 becomes 4, 4 becomes 8. These early cells remain totipotent. They are identical. If you were to separate them at this stage, each one could grow into a separate human being. This is how identical twins are made. Shutterstock explore, but soon a choice is made. About 5 days after fertilization, the embryo becomes a hollow ball called a blastocyst. Inside this ball is a small cluster of cells called the inner cell mass. These cells are no longer totipotent. They cannot make a placenta, but they can still make every single cell type in the human body. We call these pluripotent stem cells. Pluri means many. These are the famous embryonic stem cells. They are the shapeshifters. They are sitting on the edge of a cliff, looking down at a landscape of possibilities. They can jump and become a neuron. They can jump and become a muscle fiber. They can jump and become a white blood cell. But how do they choose? How does a cell with the potential to be anything decide to become just one thing? This is the process of differentiation. Differentiation is a journey of restriction. It is a journey of commitment. Imagine a student entering a university. In the first year, they can major in anything: history, physics, art, biology. They are pluripotent. But then, they declare a major. They choose biology. Suddenly, the history classes are closed to them. The art studios are locked. They have narrowed their path. Then, they specialize further. They choose marine biology. Now, the botany classes are closed. Finally, they graduate as a specialist in deep-sea coral. They are experts, but they can no longer be anything else. In the cell, This process is controlled by the gene regulation we discussed in our last lesson. The pluripotent stem cell has all its genes open. The library is unlocked. But then it receives a signal, perhaps a chemical whisper from a neighbor or a gradient of protein in the embryo. This signal tells it, become skin. The cell obeys. It begins to methylate the DNA for brain. It locks away the books for liver. It shuts down the genes for heart. Simultaneously, it ramps up the production of keratin, the protein needed for skin. It changes its shape. It flattens out. It connects to its neighbors. It has become a specialized cell. It has differentiated. Once this decision is made, it is usually permanent. A skin cell cannot suddenly decide to become a brain cell. It has committed to its fate. However, the story does not end with the embryo. You still have stem cells in your body right now. We call them adult stem cells or somatic stem cells. These are no longer pluripotent. They are multipotent. Multi means several. Their potential is limited to a specific family of cells. Think of the bone marrow. Deep inside your large bones, there is a population of hematopoietic stem cells. They are the mothers of the blood. They can divide and become a red blood cell. They can divide and become a B cell for the immune system. They can divide and become a platelet for clotting, but they cannot become a brain cell. They are restricted to the blood lineage. These adult stem cells are your fountain of youth. They are the reason you can heal. When you cut your skin, stem cells in the deepest layer of the epidermis wake up. They divide rapidly. One daughter stays a stem cell to keep the reserve full, and the other daughter differentiates into a skin cell to patch the wound. Your gut lining is replaced every few days by stem cells hiding in the crypts of your intestines. Your muscles have satellite cells, sleeping stem cells that wake up after a hard workout to build new muscle fibers. There is a profound beauty in this system. Your body is a mix of the old and the new. You have neurons in your brain that have been with you since you were born, holding your oldest memories. And you have blood cells that were born yesterday, carrying oxygen to your lungs right now. You are a tapestry of varying ages, held together by the constant renewal of your stem cells. But science has taken this story one step further. For years, we believed that differentiation was a one-way street. We thought that once a cell became a skin cell, it could never go back. But in 2006, a scientist named Shinya Yamanaka discovered a way to turn back the clock. By adding just 4 specific transcription factors to a mature skin cell, he was able to unlock the methylated genes. He opened the library back up. The skin cell forgot it was skin. It reverted back to a pluripotent state. These are called induced pluripotent stem cells, iPSCs. It is like taking the marine biologist and turning them back into a freshman student. This discovery changed everything. It means that the potential of youth is never truly lost. It is just locked away. It suggests that one day, we might be able to take a cell from your own skin, turn it back into a stem cell, and then guide it to become a new heart muscle patch for your heart, or new neurons for a damaged brain. We could heal the body using its own potential. As you lie here, floating in the dark, consider the potential within you. You are a collection of trillions of choices made by trillions of cells. Each cell in your body has accepted its role. The rod cells in your eyes have accepted the duty of catching light. The beta cells in your pancreas have accepted the duty of making insulin. They do not complain. They do not wish to be something else. They serve the whole. And yet, hidden in the quiet corners of your marrow, in the crypts of your gut, the stem cells wait. They are the watchers. They are the reserves. They are ready to step in whenever there is a need. They represent the eternal possibility of renewal. We have completed our curriculum. We started with the hard, cold definition of the cell theory. We moved through the machinery, the energy, the code, and the controls. And we end here with the mystery of potential. The cell is not just a machine. It is a story. It is a story of cooperation, a story of sacrifice, a story of memory. It is the story of life, playing out on a stage too small to see, yet large enough to contain your entire existence. The machinery of your body does not need your supervision. It has been doing this for billions of years. The DNA is replicating, the ATP is burning, the signals are flowing, the The stem cells are waiting. You can let go of the controls. You can trust the wisdom of your cells. Let the golden sphere of the stem cell fade gently into the white light. Let the white light fade gently into a soft, comforting darkness. The lesson is over. The work is done. Sleep now, in the care of your own miraculous biology.